All right, so today we're going to be doing, where is everybody? I only know a couple people that are not here right now and why they're not here, but it is what it is, I guess. So we're going to be doing chapter 15 and 16 as kind of like a lecture melded in with Kahoot questions. So we'll be basically like going through the lecture because just to kind of restate again, like I don't feel like enough people are watching the lectures. And what I mean by watching lectures, actually watching them and engaging with them. Like how many of us just sit there and they're on? That's not acceptable, especially not for 15 and 16. So we're going to do it this way for chapters 15, 16, and probably 17 and 18. We'll see. But we won't be doing that for chapters that unit 4. The reason we're not doing that for unit 4 is because you guys have presentations. I can't take away from that time. So what we'll do on unit 4 is you guys will have your presentations in whatever time you need for the presentations, and we might play Kahoots after that but we might not. It might just be presentations during unit 4. It just kind of depends on how that time ends up being spent on the days that we're presenting. So, but whatever. So we're doing chapter 15 and 16 today. I have incorporated the lecture slides into a Kahoot. And so there will be random questions. Usually the questions have to deal with whatever the slide was previously. And it kind of also lets me know, were you paying attention? Because I literally am pulling the question from like the slide or two before it. So we'll see. But yeah, I mean, and so 15 and 16, 15 is some basic information, but 16, if you can't wrap your head around 16, you're not going to do well on the test is pretty much how it's going to go. So we got to take some time on 16 to do our explanations and walking through immune response and stuff like that. So there's this, we're doing this like lecture based stuff so that you guys don't have to rely as heavily on like watching the recording. So we're not going to miss anything that way. The other thing is that when I do our weekly review on Thursday this week, I am not going to do like what I usually do. What I'm going to do is literally the whole review will be me walking through the immune response. I'm going to draw it out on an interactive whiteboard on Zoom, like the Zoom whiteboard. And so you guys could even like draw things too if you wanted to, but I'm not saying that you have to, because I'm still going to record it. It'll still be a recording of me drawing the whiteboard, but it's like me drawing like the process. Like, okay, first we'll go through the innate immune response, and now that we have the innate immune response, you can't have the adaptive response without the innate response, because it relies on some of those antigen presenting cells and signaling and stuff like that. So it's basically going to be a walkthrough of all these concepts from scratch. So that'll be the idea of what to expect for the review this week. So there's no like slides or anything getting into that. When the exam's on, I have no idea what day the exam's on. That sounds right. Yeah, I think you're right. Yes. Because it's this week is 15 and 16. Next week is 17 and 18, and the exam would be I think that's the 10th. For unit 3. All right, we ready? Everybody make sure we're logged in. If you're online, log it in. Let's see how many we have in here. 1, 2, 3, 4, 5, 6, 7, 11 in here, and then 1 online. Out of the 20 people that are in the class.
Kylie is online.
Yes, yeah.
Okay.
Let's see who is showing up on here. Yeah, it's Kylie. That's their choice, man, right? Especially, like, I don't think it could have been clearer about 15 and 16 being the deal, right? Okay, just saying, like, I hope I'm not imagining I'm saying the thing. Okay, it's fine. All right, so we're gonna do this. Get into this. So this was, we got it. This is our learning objectives. I'm not going to read through them, obviously, but just reminding you guys that I do test from the learning objectives. So if you can take the time to actually understand the learning objectives, this is the level of understanding that I would be expecting. Can you actually explain the major steps of phagocytosis? Not just list them. That's a different thing. Can you explain them? Can you explain me how phagocytosis works? Can you explain the role of a mononuclear phagocyte system? Not define it. Can you explain it? So that's another thing where I will look at concept maps, and I'm like, hey, make sure your concept maps address the thing. And you guys know by now, I'm not super picky about a concept map, right? You have the general content on there. You're putting some effort to put content on there and relate it to each other. almost everybody's getting full credit on that. I'm not super picky about it. I cannot tell you how many students just submit me an outline that ChatGPT made. I'm not crying over it, but I bet you're crying over your grade that you get on the exam later on. If you do something like that, your choice though, your choice. But if you're gonna do it and I can't prove that ChatGPT did it, I'm not blind. I know what it looks like when ChatGPT makes an outline and you just copy and paste it into Word. And I'm telling you, I can't tell you how many people are doing that. Probably at least a third of my students do that in all of my classes. So it is what it is, right? But if we're doing this and looking at it this way, most of the time when we're submitting stuff, like it'll have just like, you guys will make a concept map and it'll just like say mononuclear phagocyte system for cells to move around on or whatever. And I'm like, does that really explain to me its role in the immune response and stuff like that? So when you're studying, relate it to what the content is about, be able to go back and explain all those things to me. This doesn't look like a lot at face value. This is literally it for chapter 15, right? This is all of our concepts for chapter 15. It doesn't look like a lot at face value, and it's really not at face value. So if I can stand up here and explain to you everything from chapter 15 in an hour, it's really not that much content. But being able to break it down, explain it back to me, the work behind that, it takes work, it does. And so if you're skimping on that, and I'm not saying you are, because you guys are here and showing up, you're probably not the ones that would be skimping on it. But if you will feel like you're skimping on that, then you might know what you need to go back to and, when you're studying, readdress. So just be aware of that. My nose, I changed my nose ring, and it's like my nose has to readjust to the vibrations or something. I don't know. Anyways, it really tickles right now. All right, so this is a good overview of basically like the setup of our immune response overall. We have our innate and our acquired immunity for our host defenses against invading microbes or anything foreign that could be seen as foreign from the outside. So we have our first line of defense, which is basically our physical and chemical barriers, as well as our microbiome that provides microbial antagonism. We've kind of already learned a little bit about the microbiome and the microbial antagonism, but the physical and chemical barriers, they're obvious ones. The fact that you have keratin as a waterproofing protein that prevents microbes from getting in directly through your skin and just like living there. We actually already know how effective that is as a barrier because somebody gets a cut. We're worried about microbes getting into the deeper tissue and growing. They can't necessarily survive on the literal surface of your skin. They have to get a little bit deeper in order to live. Chemical barriers would be things like your stomach acid. For example, whenever you eat something, a lot of microbes just can't survive that stomach acid at first. Yes.
What about the blood-brain barrier?
The blood-brain,
a physical barrier?
It would be a physical barrier, yes. So, and we'll talk a little bit about that in Unit 4 when we talk about our nervous system diseases. It'll talk about the blood-brain barrier, but it's quite literally a physical barrier. And it is because the junctions between the cells in your vessels that go into your nervous system tissue are tighter junctions, and not as many cells can actually squeeze through there. It's also the receptors that they use and the signals that they send to allow things to cross over into the brain. It's trickier for molecules and cells and other things to cross into the brain. The blood-brain barrier is a very real physical thing, and it's also gated by other things as well.
So it's kind of like a little bit of, it uses a little bit of chemotaxis and then a little bit of just physical.
Yes, and so that's true in all of our innate response or all of our white blood cell responses. It's just that there's more availability to get out of the bloodstream and into the tissues everywhere else, except for your central nervous system, which is a good thing and a bad thing. A lot of what we're going to learn about today in your innate response, we're talking about inflammation. And your body, you're a multicellular organism. You can afford to lose some cells in the battle, and then you'll regrow them and you'll be fine. Your tissues can survive, but can your nervous system survive that? No. Your central nervous system, once damage is done to your central nervous system, is permanent. You're done. So we can't allow inflammation to happen in your brain when it's uncalled for, because it can literally damage your nervous tissue to the point that you won't survive it. So that's why the blood-brain barrier exists, is to protect you, because that's your core, if it can't,
literally just
that's it. Yes. So that's it. So we have to have that as a physical barrier there that prevents our white blood cells. It prevents your own body from being able to go in there to defend against things. You have your own type of immune response in your central nervous system, a little bit different kinds of cells that are relegated and specific to the nervous system. And you will have some immune response there, but it's meant to be much more dampened. And yes.
For our first line of defense, would that be our mucus membrane in our nose?
So yes, so even like mucus production in any of our respiratory tract, whether it's in your sinuses, in your throat, and in your lungs themselves, we have mucus production, and that causes microbes to stick there so that they can't make it in. We have, production of other fluids, like we say mucus, but also whenever your mucus gets really liquidy and you have a runny nose, it's trying to flush out things that could be irritating. Basically, your body says, hey, something made it in here that's triggering like histamine and stuff like that. Histamine causes, vasodilation and depends on what we're talking about, vasodilation versus vasoconstriction. The blood coming in, we want more of it, and we want less blood coming out of the area. And that causes swelling, which we call edema. And then that would lead to basically like buildup of white blood cells in there. And we're going to create exudate, which is going to come out as a runny, watery nose. We're trying to flush things out, whatever's causing that histamine response and all of that. So that's all kind of relating to that. But that mucus, the runny nose, all that act as physical barriers as well as chemical barriers when we start talking about the chemicals that are in the mucus or the chemicals that are in your saliva, for example, like lysozyme. Lysozyme's in your saliva, in your tears, and in your sweat, and it breaks down peptidoglycan. So it's very like wide chemical breakdown of any bacteria that have peptidoglycan. Yeah, so very useful. So that would be in our chemical barrier in our first line of defense. So very general response in that first line of defense. The second line of defense, if something gets into your body, we have our innate response. And that's what we're going to be talking about the most today. So it's going to be like all the white blood cells that don't do specific jobs in immune system stuff, but general jobs. I consider them like your basic like soldiers on the ground. Okay, so these will be your neutrophils, this will be mast cells, even macrophages and monocytes will fall into this category. These are the guys that can say you are foreign. I can say that you are a person, but I don't know who you are. You know what I mean? Like we were saying in lab, like it's like that. So I can tell you are a person, but I can't tell if you are Deontay or Maddie or whoever. Like it doesn't, like it can't tell. So our third line of defense can tell. And so in the beginning, we're like, okay, a person is here. We need to have like general, like, anti-person, anti-invader response. But once they are like, okay, we have identified some parts of our invader, we'll bring them to our third line of defense, our lymphocytes. And our lymphocytes, well, whatever lymphocyte, like we said, we have 8.6 billion different ones. And so that lymphocyte that only sees Deontay sees a piece of Deontay and says, I've been waiting to see this piece my whole life. They'll proliferate and then differentiate into different types of T cells or proliferate and differentiate into the plasma B cells and all this other stuff that we're going to talk about in chapter 16. So that's the big overview. But you can't get third line of defense without help from the second line of defense. And they often collect the parts to help us like react to the specific parts. They also keep things at bay until we can get third line of defense activated, because that can take like a week or two in order to get that going. Yes.
So I know that like pediatric patients won't often have like really good immune systems. What time, what period does the immune system normally start kicking in for like pediatric patients?
Usually your immune system is going to be at full force between 6 months to 18 months of age.
Okay.
Once you're that age, it's like gets a little stronger and stronger until you are like, I want to say like probably, I want to say 4 or 5 years old is like when it's at its strongest.
Okay.
And then pretty much from there, it'll stay pretty strong until you hit puberty. And then you're pretty much screwed after that. Your immune system starts sucking after puberty. So yeah, it'll be at its height between about 2 years to 3 years of age and about puberty. And so yeah, if you think that you have a stronger immune system because you're an adult, that's a disillusion. That's not true. But your immune system has been trained your whole life against all these things, and you've built up your defenses, and now they're saved. And that's why that perception is there. It's just that kids haven't seen a lot of the new things. And so we have memory to a lot of things as adults that's been activated throughout our whole life, but kids are seeing those things for the first time. And so they're having to get their memory, and it takes a week to build memory. Yes.
One other question, and then I'll like stop with the
Yeah.
questions for a second. Is it also true? I think I learned this in like middle school or something, but I heard somewhere that 2 organisms coming together and having an offspring, it basically combines their immune system, and then it makes a stronger immune system for that offspring. Is that
It
true?
can and it can't, right? So, I mean, this is true for all of our genes. So all of our genes, just a real quick genetics lesson, okay? you have 23 pairs of chromosomes, right? So you have, I don't know, we'll draw 4. I mean, let's say we have 23, but I'm going to draw 4. And let's say you got 4 from the mother and 4 from the father. All right, so this would be chromosome 1, 2, 3, 4, and the father had chromosome 1, 2, 3, 4. And you might be sitting there saying, like, no, the mother had a pair. You're right, the mother did have a pair. But when you make an egg, if you need pairs to come together, it can't have pairs in it. So when you make an egg, well, you might have had pairs of chromosomes. If you think of them as X's, you're also wrong. They're not X's here. Okay, this, where your pairs of your chromosome, this would be the actual body, your actual somatic cells, your body cells, okay? And then these, when you're going through mitosis, each of those pairs turns into an X. Whatever, this isn't I'm drawing because they're attached at the center. So this is what's actually happening. So each of this. Okay, so this would be all, like, again, you have 23 pairs, right? And one of those is your sex chromosomes. So whatever, so this is in your body when we're having this. This is what happens in mitosis. In meiosis, when we're making our sex cells, you would have one of each of these guys because they're going to come together and make the total. You get what I'm saying? Because you can't have 23 pairs coming up with 23 pairs. That's too many chromosomes. You can only have 46. So meiosis, you would only get one. Now, this one came from the mother, and this one came from the mother's mother, and the mother's father, etc. So we get the big picture there. Okay, so once we get to the gametes, the actual in the gametes, again, this is what it would look like. Okay, so again, an egg and a sperm come together, and then that chromosome 1 from the mother and the chromosome 1 from the father. It is not as simple as you get them and now you have different combinations of genes. It doesn't end there, okay? What happens is after you have fertilization of the egg, I wish I had different colors here because it'd be easier to draw it, but basically like chromosome 1 of the mother and chromosome 1 of the father will cross over with each other. And then they will attach and then create new chromosomes like this. And so you have crossing over of chromosomes and then new creation of different genetic information on each chromosome now. So you don't have the chromosomes from your mother and the chromosomes from your father. You had a weird amalgamation in your body of the crossing over of the different combinations of these guys. Okay, so as that happens and we have increased differences between people, again, no crossbreeding, because this is the problem. Well, I mean, inbreeding, you get what I'm saying? No inbreeding, okay? Because if you have inbreeding, you're just going to have repeat of the same genes. There's not as much variability, not as much crossover, and then your offspring don't have new genes compared to the parents that have the same freaking genes, and you're not getting enough variability in population. So when we say, yes, okay.
So it's kind of like a happenstance, it depends.
Okay. And that is why, whenever any parent gives birth to a child that isn't an identical twin, what is the likelihood? I mean, a parent, like, I look at the Duggars, right? Okay, I'm just saying because they've had a lot of kids, okay? You know what I'm talking about, but it's like 16 and counting or whatever it is. Yeah. So those kids are not identical twins, right? They had like what we would consider just like normal, like one kid at a time. None of those kids are exactly the same, right? Because of this. Because their genes are crossing over and all this stuff is happening. And whenever they came together, the mother's mother and the mother's father, they crossed over. And then you're not going to get any of the same chromosomes coming out of the gametes that way. So this crossover happens usually during the gamete formation and can also happen after fertilization of the egg and stuff. So it's usually gamete formation when we see the most crossover happening. Whenever you're forming your eggs or you're forming your sperm, we'll get that crossover happening between your existing guys, but it can happen after fertilization and all sorts of stuff. So you can see there's a problem if you have, 3 of a chromosome, for example, because then just splitting up stuff doesn't work the way that it's supposed to. All right, but anyways, that's like the most watered down genetics that I'm going to give you. It's a lot more complicated than that. But yes, so when we talk about genes, like if this chromosome has a gene for eye color and that chromosome has a gene for eye color, well, that's not going to change. Like you're still going to have a gene from the mother and a gene from the father that are the gene from them. Those aren't going to change. But the gametes will have a different layout basically than what the parents' body cells were, and then the egg can have a different layout than that. And so the predictors there's a lot of variability that can come into play because of the genetics of making eggs and sperm and things like that. So it's meiosis. Now you can sit there and say, thank you for me not having to teach you meiosis and how all this works. Because I mean, this is like, there's a whole entire semester for just genetics. And if you're going into study genetics, this is like, I wouldn't be able to explain that level of stuff. And it's been a long time since I took genetics. But anyways, yes. So if we talk about the likelihood that you're going to have greater immunity because your parents, coming together and making a new person, and they get stronger and stronger. It's stronger because of just genetics and how it works. And that if your parents were related, if they were brother and sister, for example, in the example of incest, we're talking about close, genetic relations there. And yeah, you're not going to have as much variability, not as many crossover events, not new stuff being introduced. And that increases every generation that you have incest happening, which used to happen quite a lot back in like the dark ages, whenever kings would just only breed within their families and stuff like that to keep their bloodline alive or whatever. So that's why incest would be bad. And then you're going to see not just abnormalities in your immune system, but abnormalities in all aspects of the body. And so that's just because we're not having new variation of the genes in. Okay.
Immunity more so from breast milk, vaginal canal.
In the beginning of life. So in the very beginning of life, like I said, you don't have an immune system until about at least minimum 6 months of age. Mm-hmm. Some people that's longer. And so we usually put off immunizations until 18 months, just to be sure that the child has an immune system, just has one that is actually functional and can deal with the antigens that we're going to present them with. So that's waiting on the development of like their naive version of their acquired immunity. That's waiting on that to just exist. Yes, from the bone marrow and all those cells like multiplying and growing and then becoming, trained on how to deal with things. So there's that issue. The other side of it is we get antibodies and stuff from the breast milk, but it's not actual true immunity. Like that we only have the immunity that our mother had. So if our mother never saw a bad guy and it's our first time seeing the bad guy and the mom's first time seeing the bad guy, we're not going to get those antibodies until she gets her own acquired immune response and it's present in the breast milk and stuff like that, right? So we would have to wait as a baby on that as well.
So in that way, like let's say your mom's immunity sucks, yours isn't going to be as strong potentially as somebody else whose mom is strong.
Right. Yes. And so there is a thing like this, right? There's a thing called like a breast milk donation or something like that, where you can actually have mothers can donate their breast milk and they can freeze it and like cold storage it for mothers that like have babies that want to give breast milk but they can't give their own breast milk and can supplement with natural breast milk that has things like antibodies and stuff like that that are going to be more healthy for the baby.
So for autoimmune diseases then that are passed on from like the mother or so, is that when the gametes?
Potentially. Okay, so that's the thing too, is do we see that autoimmune diseases can be genetically linked? Yes, but it's way more complicated than that.
Like
so
activate the genes.
Yes, so it's like a lot of this stuff in our lives where you can have the genes for a disease, but environmental factors, trigger them to behave a certain way. And so with autoimmune diseases, it's usually like you have the genes that predispose you for an autoimmune disease. You're not guaranteed to get it. But if you are exposed to certain triggers for those things to get activated, then you will. That's oftentimes, and we'll talk about this in chapter 17, going to be like microbes that look like self or microbes that just happen to trigger certain types of antibody reactions and stuff like that. So there's all different types of ways that your immune system can kind of go wrong. Autoimmunity is one of those things.
Can it even be lifestyle and diet?
Yes, so it could be that too, and how healthy you are, or like how you're dealing with even being ill if you're ill with something else, and it causes some sort of inflammation process, then that can trigger autoimmunity just by certain signal molecules being there. Stress is probably one of the biggest triggers of your basic overall health. being poor, just being obvious, just being clear here. And we kind of know this because we live it on a day-to-day life. But if you're constantly tired, if you're constantly having issues with fatigue, if you're sore a lot, and if you're having other issues with your health, it's probably related to the amount of stress that you're dealing with, because stress is an inflammatory process. The amount of cortisol that you're making, the amount of other hormones that are involved in the stress response can basically trigger other inflammation and things like that. Your body is meant to have that basically to help you deal with stressors that are meant to be temporary, not chronic, like we have now in our lives. Yes.
The last one I'll say, sorry.
You're good.
So when, I don't know if you've heard or your thoughts on when people say that like trauma can be inherited, is that more so just saying like nervous system dysregulation, stress response, inflammation response, and maybe those genes being more, you're more predisposed if it's been like generations of...
That is a whole other conversation.
Okay, we don't need to get into it.
Yes, so trauma cannot be related, period. Like cannot be inherited, period. Like it can't, it's not. But if your mom was in a traumatic upbringing and suffered child abuse, she's more likely to be a child abuser. That is facts.
Right.
Like the numbers show that. And so we have that. And we have that issue of like, and that doesn't matter. Like if, like, for example, my mom was a child abuser and her mom was a child abuser, and it goes way back, and like, whatever. Did her mom want to be a child abuser? Probably not. She probably knew that her life was tough and she was under stress. So my grandma being, very emotionally and physically abusive, stuff like that. I'm just using my personal example. Believe me when I tell you I've gone to the therapy and everything like that. But like for that, right? And then now my mom doesn't know how to deal with stress, because who teaches you who's your model for how to deal with stress and life and manage things? It's your parents. And so, instead of my mom going to therapy and trying to learn how to break the cycle, my mom just does the same things that her grandma did, or that her mom did, but my grandma did, and whatever. And even though she's like, your grandma was, a horrible person too, and who was I supposed to learn from? Whatever. I'm like, well, I went to therapy and I'm changing this cycle. And I look at it, I'm like, I'm not going to be like you. But that ideology comes from the same thing. Well, my mom believes that she is that way because it's in her genes to be that way. That's false.
Yeah.
That's a learned behavior, right? And so it's stuff like that. So that gets more complicated. And does she have genetic predispositions to be more reactive to things? I don't know. And so that's a little bit trickier.
That's
yeah.
more into like behavioral psychology.
Yes, and is that related to genetics too? Maybe.
Absolutely.
Yes.
Yes.
Yes. So right. So that's really
I think
that's
more
tricky.
that side of it, less the behavioral side.
Yeah.
Of it, but yeah.
Yes. So that's a tricky one. But anyways, we're happy to have that conversation with you if you'd like to get deeper into it. As somebody that's been through a lot of therapy, yes, I can tell you that sometimes it's people lean on stuff like that. Like clearly you can see that in the family line, there's all these, trauma and whatever. Yes. And they use it to justify their behavior. Let me tell you, when none of my therapists agree with it.
Abuse period is
Yes, agreed.
So kind of,
Yeah.
when we're talking about the second line and third line
Mhm.
combined, basically, is that kind of the second line is we want to, our APCs are coming in to show?
Yes, and we'll talk about the APCs, yes, but that's exactly right, yes. So when we get into the acquired immune response, you'll see that our lymphocytes, our B cells and T cells require that we be presented antigen by specialized cells through specialized flags, basically. And those are going to be our antigen presenting cells that are basically like, we present antigen. That's what we do. We don't say this is a person alert. We say this is Deontay alert. And so how do we actually use that to awaken our B and T cells? So many of our antigen presenting cells are going to be in that innate response. We generate them in the innate response, and then they will go on and activate the adaptive. So we'll get to them. But first we got to introduce ourselves to the innate response. So where on the chart would nostril hairs fall? Don't think about nostril hairs being part of your immune response, but where on that chart that we just looked at would you expect nostril hairs to fall? Just drop your pins. Go away.
Yeah.
Yeah, just touch it somewhere. Touch it or click on an area near where you think the answer is.
This is the past. I saw like drop your pin too, but it was like.
All right, timer's up. We got several first line defenses correct, but specifically down here in the physical, that's absolutely correct. So good job on that. Yes, definitely innate, definitely first line of defense, and definitely physical barrier, because it actually is physically trapping like particles from getting into your nose. Now, if you're someone like me where you trim your nose hairs, because I have this nose ring, and so I have to trim the nose hairs or else things get even weirder. You think it tickles now. So, but yes, so I am removing some of that barrier into my sinus cavity and potentially setting myself up for being more likely to have things like sinus infections or something like that. Now, how real is that in somebody that has an otherwise healthy immune system? It's pretty negligible. But it still is one of the types of barriers that we have to keep microbes from getting into our body. So let's talk about some chemical defenses. We already talked about stomach acid, digestive juices in the intestines, including bile, which has antimicrobial properties and is alkaline. Semen even also has antimicrobial chemicals in it, and the vagina has an acidic pH as well as microbiome there. Microbiome themselves offer microbial antagonism, as well as by creating the acid environment themselves because they're fermenters. So lactobacillus is the #1 species, and we'll learn more about them when we get into chapter 24 when we talk about the microbiome of the genital tract of both women and men. We talked about lysozyme. It's an enzyme that hydrolyzes peptidoglycan. That means breaks it down. Hydrolysis is something that we're going to be very familiar with after our labs get into gear. But we learned about hydrolases when we talked about chapter 10 in our different classes of enzymes. That hydrolases are basically enzymes that break down other molecules by adding water, and they split a water molecule and basically like stamp on like an OH or H group onto the ends of molecules and stuff like that. So it hydrolyzes peptidoglycan. We also have salt and low pH. We talked about microbial antagonism, basically microbes taking up our stadium seats so that the bad guys can't sit down, but they also make antimicrobial peptides, things like that. They make exotoxins, exoenzymes, things that can actually inhibit the microbes. They're not just taking up the stadium seats, but they're also using it for nutrients in the environment as well. Okay. Also, our microbiome trains our host defenses as far as what and when and where and how our body should, attack something. So we have E. coli and stuff in our gut right now. Why is our immune system not attacking it? Because our immune system has basically decided, whole organisms that are sitting in your gut are fine. If those organisms get outside of your gut, we got a problem. So it knows when and where and how. And so your gut microbiome kind of trains your body on that earlier in life. If you don't maintain a good gut biome or don't keep your gut biome happy, that can actually affect your immune system because of that training aspect going on there. In the genitourinary tract, we have a trickle of urine that flushes out things in the urethra, as well as vaginal secretions that help slough off with the scales to desquamate the cells of the vagina, to desquamate the cells, to remove any microbes that could be attached to the vaginal wall, as well as the flushing action of the mucus and stuff in the tract as well. In the respiratory tract, we have nasal hairs, we have mucus, we have the ciliated epithelium in your respiratory tract as well. That basically is like cilia constantly moving things out of your lungs. So we have fluids and mucus and things like that in your lungs, but the cilia are constantly moving it out. It's why you cough. It literally is why you cough. It's to help all that stuff move out even faster. But it's always trying to move things out. When you get pneumonia, there's so much fluid, the ciliated epithelium can't help move it out anymore. It just settles in there. And so what happens is the microbes are quite happy with that. And they end up living there, and they can actually damage, coughing and things like that can actually damage the cilia, and we no longer can move things out. When we talk about whooping cough, if you've ever heard somebody have a cough, like we know if you drink water and you inhale it, you get that type of a cough where you're like, that's a whooping cough, okay? But that doesn't mean you have the disease whooping cough. If you have the disease whooping cough, which is caused by the bacterium pertussis, it's Bordetella pertussis, okay? Bordetella pertussis. If you have that, you will have that cough for, it's called the 100-day cough. So we're talking 2 to 3 months worth of coughing, like constantly. That's the whoop of whooping cough. And it damages the cilia. So then you're susceptible to secondary infections. So now you don't just have that bacteria, but even if your body clears that bacteria for months afterwards, you don't have cilia to clear out other infections. So you get other infections. And so you might be sitting here being like, man, I'm glad I am, immunized against pertussis. Yes, well, little babies in the NICU aren't, and people don't tend to keep up with their Tdap vaccines, which is where you would get your tetanus, diphtheria, and pertussis vaccine, unless you step on a nail and you go to the, urgent care and you're like, have you had the vaccine in 10 years? You know the drill. So if you haven't, and you're an older person, you might have whooping cough and have very mild symptoms, or even just be a carrier of the bacteria with no symptoms at all. But the little babies get that cough, and they can die very easily from that. It's still not uncommon to see in like NICUs and things like that nowadays for pertussis, because they don't have their own immunity. Big thing there. Mucous membranes, of course. So the mucus itself is a flushing action. We can also have antimicrobial peptides and things like that. But because the whole reason we have wetness in the mucous membranes is because we don't have the keratinized layer to protect those surfaces. And so they're more susceptible to infection. So anything that is a mucous membrane is more susceptible to infection because we don't have the keratinized skin layer to protect us. So they are moist, they are permeable, but we always have flushing of fluids, usually on our mucous membranes in some degree or another, even blinking and tears and things like that in our mucous membranes of our eyes and stuff like that. So, all right, moving on, hair follicles, skin glands, flushing with sweat and stuff like that. The keratin is a waterproofing protein, and that creates a barrier to keep like the different layers of skin and their own functions. If you have the epidermis versus the dermis, and talking about stuff like that, then there is quite a difference between the type of infections you could see in each of those layers, and that whole epidermis is basically creating that protective barrier for the dermis and the tissues underneath. All right, cool. So moving on to immunology stuff. So that was it for our primary defenses. That's like our body's basic. Do those things have other functions besides protecting from microbes? Of course they do, but they also can act those things as like a secondary function. So immunology is the study of our ability to recognize anything foreign and then react to it. We have white blood cells. White blood cells are also known as leukocytes. So if you ever heard of leukocytes are white blood cells. Leuko is the white part and cytes are the cells, okay? So any white blood cells, all of your white blood cells, leukocytes, okay? They're trained to recognize self and non-self. Some of them, this is very general, like saying this is a person versus the table. Sometimes it's like specific people, okay? So it just depends on the cell that we're talking about. And they can tell the difference between self and non-self. And this is probably the most important thing that they have to be able to do, because otherwise we would be constantly attacking self. And so we have that training happening early on in the development of your body in the uterus. So we're often training that even before you're born. And even then, again, the first few months after life, okay? Antigens, we also call them immunogens. These are any type of specific molecule that we could potentially react to. So these are the markers that let me know that it's Deontay as opposed to Maddie. Okay, so those are markers that tell us the difference between things. They're often found on surfaces of cells or viruses. They're usually proteins or sugars, but they can be any molecule that has a shape that we could potentially interact with appropriately. And so we now know that based on your, basically your amino acids and whether they're charged or not, or whether they have big hydrophobic groups or whatever, that dictates the folding of your proteins, and antibodies are proteins, and how they fold will dictate what they can interact with. And so we can get very specific with that. Okay, so foreign things, basically we can tell whether something is foreign and how specifically foreign it is. And autoimmune disorders basically exist because we accidentally see self as foreign. We'll talk more about that in chapter 17. I don't want to get too much further into it now, because I want to get into our basics of the immune response. Pathogen associated molecular patterns, PAMPs. And do you have to know the acronym? So, if it's bolded on here, I would probably know it, okay? So a PAMP, a pathogen associated molecular pattern. These are things that tell me this is a person instead of a table, okay? So they have a face, probably a person, stuff like that. So it's a very general thing. Now, the specific idea of the specific things that make that Deontay's face versus Maddie's face, those would be antigens. But the fact that I can see that it has a face, probably a person, that's like a PAMP. And so there are some examples of PAMPs that we'll probably need to be familiar with. Lipopolysaccharide or endotoxin. Remember that one on the surface of gram negatives? That's a PAMP. So we can see that if our body sees lipopolysaccharide outside of like an actual cell broken down bacteria, because we have it in our bodies right now in our guts, but they're whole cells, they're happy, they're not being attacked or broken down. But if we see parts of those cells, including lipopolysaccharide or peptidoglycan, floating around in your blood or something like that, your body can immediately say, this is foreign, this is non-self, and we need to react to it. So a person is here. That's what they'll be saying as a result of that. So PAMPs include the big three that you would definitely need to know. There's way more than three, but the big three that you definitely should be aware of are endotoxin, which is lipopolysaccharide, peptidoglycan, which is part of the bacterial cell wall, and double-stranded RNA. Because we don't have double-stranded RNA. We have weird folded up like tRNA, but it is not double-stranded. It's still only a single strand. What has double-stranded RNA? Viruses. That's it. And so if we see that in our body, that's another way that our body knows, like, nope, you aren't us, you're a non-self. Okay, so they have pattern recognition receptors, also known as toll-like receptors. These pattern recognition receptors recognize the PAMPs. So the PAMP is the peptidoglycan. The pattern recognition receptor just recognizes that this is one of the PAMPs. Okay. So that's how most of our innate system responds to things. So true or false, a PAMP is a molecular marker that helps the innate immune cells recognize general groups of microbes. Oh, right, I have to click on it. Here you go. Go away. This is true, very general statement here, talking about what the PAMPs do. So moving on, our lymphatic system. This is getting into the idea of the lymphatic system, which this usually surprises people unless you've taken anatomy and talk a little bit about this in anatomy, but I still feel like it's not really ingrained in us to understand that the lymphatic system is a completely different system. And so it takes extracellular fluid that came out of your blood. I want to be clear about this, okay? So everything that's in your blood can migrate into your tissues. It's how your tissues get the nutrients and the things that they need. It doesn't stay in your blood, okay? So it migrates out of your capillaries, it migrates out of your vessels and into the tissues. That's also how all of our white blood cells and all of their signals that they send get sent into the tissues too. How their platelets get there and things like that to cause clots to happen, okay? All those come from your blood vessels migrating into the tissue. And so extracellular fluid like that, with all of the signals and the clotting factors and all that, moves into the tissues and out of the bloodstream. But we do not just reabsorb that back into the bloodstream. I hate to break it to you. A lot of us think like, okay, once that fluid stuff is done, it gets reabsorbed back into the bloodstream. Yeah, eventually it does. But first things first, it goes through the lymphatic system. And so what happens is all of those fluids, like you stub your toe, happens every day, right? Maybe not your toe, but you always hit your elbow or your toe, or you grab your cup weird and you knocked your knuckle on something or whatever. Inflammatory response starts, right? And so I get bruises, y'all, let me tell you. Okay, so I get bruises all the time because I am 40 and I don't pay attention when I'm doing and have ADHD. And also I do Pilates and we do a lot of like kneeling on things and I get bruises. So what happened to get those bruises? Did I rupture some capillaries? Yes, but we also had fluids and things coming in there to say, hey, we need to heal these tissues. All of that stuff gets sent into the tissues as exudate, okay? And it does not come back into your circulatory system. Yes, and so all of those fluids and all those cells coming in there to repair things and send signals saying we need to repair things that are saying we need to vasodilate, or we need to vasodilate, or we need to vasoconstrict, depending on what's going to the area. Vasodilate what's going there, increase blood flow there, but decrease blood flow out of there because we don't want microbes taking that as a highway back into the tissues or whatever. Go ahead and patch things up with platelets and fibrinogen and all these like markers in our blood and whatever, and do all that. But it does not come, once we're done, we do not come back into the bloodstream. We come back through the lymph, which is a different system, different vessels, okay? Why is that? Because if you injured yourself, even if you just stubbed your toe and you didn't open any skin or anything like that, your body wants to play it safe. We had blood open up into an area and things open up into an area where it doesn't normally go into. So we don't want to bring back with us microbes straight into our circulatory system, which is a sterile environment. It's an open stadium. We don't want to bring that back. So we go through the lymph system. It is literally just a pathway through which you are filtering everything from the extremity back before it can rejoin the bloodstream. And it's quite literally what it is. What if something made it past the lymph and made it back into the bloodstream? Would your body be able to circulate it and then help filter it out? Yes, you have one more organ after that that is specialized for that, and that is your spleen. So your spleen will filter things out from the blood if it makes it that far. But you have thousands of lymph nodes throughout all the tissues of your body. We think of them being aggregated in our armpits, in our throats, even in our gut and in our groin and stuff like that, because we feel them and palpate them because those are areas where they're aggregated the most. But you have them throughout your entire body everywhere. And they're super, super tiny. If you've ever seen a lymph node, they're about the size of this eraser, the white on here, okay? That's how big they are normally when you are healthy. If you have an infection, they get big where you can actually feel them. And so that's what actually is happening there. Now, okay, so that's a drain off system. Lymph itself is just plasma, like it's the fluid that came out of your blood. So your red blood cells only come out of there if you have a break in your capillary and stuff like that. So not your whole blood that's coming out typically unless you have an injury. It's the fluid, it's the signaling molecules, it's the clotting factors in the white blood cells. So lymph is all of that coming back in to the body. And it's basically going to be going and being filtered out through your lymph nodes. Lymphatic vessels basically come along a very similar pattern as what we have with our capillaries and our blood vessels and things like that. They're from all over the body, throughout all the tissues and everything, because you can get injured anywhere in your body and still need to filter back in before we rejoin. Lymph moves in only one direction only, okay? From the extremity back to the heart. So not from the heart to the extremity. Lymph, that's how your blood flows. Your blood flowed to the injured area. Now you're injured. We healed up everything outside of the bloodstream. Now we need to rejoin the body. We need to rejoin the bloodstream, but we can't rejoin it directly. We have to go through the lymph first. And so we'll come back into the lymph vessels and then circulate through all of our lymph nodes in order to be filtered out. Lymph is only transported by the contraction of your skeletal muscles. Your heart beating doesn't pump your lymph. That makes sense because lymph is kind of like a weird one direction street. It's not being forced by your heart pumping. And then your heart doesn't pump things back into you. We already know your circulatory system. The whole reason that your veins run back to your heart is because the arteries were pushing away from the heart, like that pumps itself. So lymph is not contracted by your cardiovascular system. It's contracted, it's moved by your skeletal muscles, which is why you'll see anytime you go to a hospital and somebody's in a bed for longer than a few hours, we put those things on their legs and now that like squeeze their legs periodically. And it's also why we want our patients to get up and move as soon as possible after surgery. We don't want somebody to be bedridden if we can help it. Even if you are really, really sick, I highly, highly encourage you to get up and walk every hour or so, just go to the bathroom, tootle around in the kitchen for a little bit, just a little bit of light movement of your skeletal muscles, because your lymphatic system only moves because of your skeletal muscles. So we're really already getting into cool stuff here. All right, so here's an image of your circulatory system versus your lymphatic system, which is in the green. And again, if you get injured in your foot, that'll move back up through all of these lymph nodes. That'll be between like that foot and the heart. And then we'll rejoin the venous system before coming back to the heart and like instituting all those fluids back into the circulatory system. Okay. So when we talk about lymphatic drainage, and like people are doing all these gua shas and all this like lymphatic massage and stuff, is that actually helping with your lymphatic system moving? It is. Yes, that's a real thing. Yes.
Especially after breast cancer.
Yes. Yes, massive edema because of the loss of lymph nodes, because of and scar tissue impeding the movement of that. So how's lymph transported through the body, everybody? All right. So there's two choices here that'll be correct. Good so far. Good, good, good. All right. So from the extremities back to the heart by a skeletal movement. Excellent. So our lymphatic organs, obvious ones will be things like your lymph nodes. We think of those as being primary lymphatic tissue, but it is not. Our primary lymphatic organs are going to be your red bone marrow and your thymus. This is where our white blood cells come from and where they are trained. The secondary lymphatic organs, like the lymph nodes, the spleen, the salt, malt, and galt, which are basically associated lymphoid tissues, so skin-associated lymphoid tissue, mucosa-associated lymphoid tissue, or gut-associated lymphoid tissue. This is tissue that isn't like in node form, but it performs a similar function to the lymph node stuff, as the lymph nodes do. Basically, the spleen, lymph nodes, and the alts all basically serve as like areas where white blood cells wait and filter out what's coming through the lymph. So this is where the filtering comes from. So think of it as this, right? You stub your toe, bam, and now you got this inflammation. We send white cells, we send, clotting factors and all sorts of stuff to heal the tissue. It can't rejoin the bloodstream until it's been screened. It comes through the lymphatic vessels and we go through lymph nodes. In the lymph nodes, there are basically like checking agents. Think of it as like customs. So everything that comes from the lymph nodes, all the cells there are checking to see, do I recognize you? And I either recognize you as self or I recognize you as non-self. And so if there's anything about you that says non-self, we're going to tag you. And that's what happens in your lymph nodes. And that's the importance of the lymph nodes, because we don't want anything from your damaged area rejoining your bloodstream if we can filter it out first. Makes sense? Great. All right, in the bone marrow is where all of your blood cells, your red and your white cells, come from your bone marrow through that process of hematopoiesis. And then your T cells are going to be basically trained in the thymus. So naive T cells mature in the thymus. The T cells are T cells because they are trained in the thymus, which is the T. Okay, that's the only thing that happens in the thymus. Your thymus, again, about here, about here. And it will start shrinking as you get older. Once you hit puberty, it basically shrinks and disappears. We kind of already talked. I'm not going to talk about the structure of lymph nodes, but they have different levels of tissue where you'll find different types of white blood cells doing different things in there. Again, that's a lot for this class, so we're going to kind of move on. Lymph nodes get larger because as you're activating, like let's say I am screening, I'm a customs agent, and I look at Deontay's passport, and I'm like, I've been waiting for you. I've been waiting for you. And so then I will proliferate. And then that whole lymph node swells because we're having that proliferation and all this signaling going on saying, we found Deontay, we found Deontay, guys, and like waking everybody up and all this signaling for inflammation and stuff like that. That's why lymph nodes get enlarged, because we have specialized cells there. If you're having an infection from COVID or you're having an infection from flu or something like that, we're not usually reacting to just one marker of those guys. We have all these different surface markers and the different parts of the surface markers. So we don't just see it as Deontay. We see it as Deontay's hip, Deontay's left eye, Deontay's right eye, and all of this stuff. We can see the different parts. And then you'll have maybe this lymph node had a guy waiting that could see his hair, and another lymph node had a guy waiting that could see his eye or whatever. And so we have several different reactions going on throughout the whole body from an infection. All right, the spleen does a similar thing. It filters the blood, not the lymph. So if it makes it past the lymph, or if you have an injury somewhere where we bypassed the lymph, then we would be filtered in the spleen. If your lymphatic system and you are otherwise completely healthy, you can live a perfectly normal life without a spleen. Are you more susceptible to infections without a spleen? You are, but you can function normally otherwise. Okay, so that associated lymphoid tissues. Again, these are just not concentrated little, pea-sized tissue. That's just diffuse tissue that has similar function associated with it. So we think that things like the appendix might also play a part in this as well. Anyways, blood. Your blood is whole blood. You have the liquid portion of the blood and you have the cells that are in the blood. The liquid portion, if we treat it to prevent clotting, the liquid portion is called plasma. If we don't, if we just take your blood, let it clot and separate out the liquid portion, it's called serum. So that's the difference there. We added an anti-clotting agent, then it's plasma. Serum is from clotted blood. So they are different. White blood cells, there's two different types of white blood cells, granulocytes and agranulocytes. So all of your white blood cells are one or the other. And what is a granulocyte? So cyte means cell. We can agree on this? Okay. So they have granules. What's a granule? Well, they can be a lot of different things, actually, as it turns out. But the real marker here is what we're talking about with hematologists. So if you go to the lab and you run a CBC or something like that, the person that ran the CBC and gave you those values was the hematologist. And so we can actually stain the cells so we can see them better. If they have granules visible, they look like little granules, like a grain, little tiny grains, granules. Then they're granulocytes. If they don't have granules, they're a granule without granules. Makes sense? That's it. So here's the differentiation here. The granulocytes are the phils. Neutrophils, eosinophils, and basophils are granulocytes. Plus one more, mast cells. Okay, so those are all of them. That's all the granulocytes. So if it's not a fill or a mast cell, it's probably an A granulocyte for the white blood cell. Okay, red blood cells are non-leukocytes. They aren't granulo or agranule. Their red blood cells are different. Platelets are not actually whole cells. They were cells that became parts of cells, and now they have a different function. And they're very, very short-lived. So red blood cells and platelets are non-leukocytes. Okay, if you are a leukocyte, which is a white blood cell, granulocytes are the fills and the mast cells. Agranulocytes. So what are agranulocytes? Monocytes and lymphocytes. So there's two. So you're like, there's way more than two up here. You're right. You ain't wrong. Okay. So there's really only two lymphocytes that I need you to worry about. Do we have natural killer cells? Yes, but I'm not going to test you on them. What I really want you to focus on are the B and the T cells. Those are our lymphocytes. And they're in the acquired immune response. So we're not going to talk about them right now. We're going to come back to them. Okay. So B and T cells are the lymphocytes. Our other agranulocytes are the monocytes and their derivatives. So monocytes are in the blood. If they move into a tissue, they become either macrophages or dendritic cells. Okay, so think of macrophages and dendritic cells as being monocyte derivatives. They came from monocytes, but once they moved into a tissue, they became macrophages or dendritic cells. Dendritic cells are a type of again monocyte derivative that we see typically in like skin, like localized in the skin. Whereas macrophages are a little bit more widely found, but we'll have them specialized in specific tissues. Like you have alveolar macrophages that are found in your alveoli in your lungs. Did you have a question, Taylor?
You just answered it.
Okay.
So they can be specific.
Mm-hmm. Yes, once they get out of the bloodstream, they can become specific to that area.
So agranulocytes, can they be secondary or like can they be innate or
So yes, so the lymphocytes are our adaptive response.
okay.
Our macrophages, though, they kind of are the go-between. So we'll talk about them in a
So the
little bit.
monocyte could be either or.
Yes, so it's tricky. So they themselves, right, so they are they're probably our most important of our phagocytes, our macrophages and our monocyte derivatives, but especially macrophages. Because they are phagocytes that can eat things up, but also recognize things as being foreign, and then I'll signal for the specific response.
Okay.
So they don't know that it's Deontay's eye, but they can say, I found this. And then a T cell can come and say, oh, that's Deontay's eye. I've been waiting for that, and then react to it and then signal the response against Deontay. Make sense? Yes. So the monocytes, they're important to signal the next step, but they don't recognize those specific things, but they start signaling with the specific
Is
things.
all they do signal the T cells, or do they help the T cells once it's been properly signaled?
They help too. So like, let's say this, like, let's say I come along and I find Deontay's eye. Deontay was like, I stubbed my toe, whatever, Deontay got in through the toe stuff, okay? And my macrophages in that area, my neutrophils will come in and they'll start eating things up. They will eat things up by recognizing paint. So they say, this is the person and ate it. This is a non-self person, ate it, whatever. But they can't tell it's Deontay yet. And they're just like eating it and then dying or leaving behind waste. Then the macrophages come and they're eating things up. This looks like a person. They eat it up. Person, eat it up. Ugh, the neutrophils left all this stuff. Eat it up. And then they're like, found all these things. Just, they don't know what they're showing off, but they found this stuff while they were eating stuff up. And then the T cells in the lymph nodes or in the periphery or whatever will come and be like, that's Deontay's eye. Everybody, we found Deontay's eye. Get ready, we're gonna attack. We found Deontay's eye, guys. That can't happen without eating up the stuff.
Yeah.
So very general eating up the stuff then leads to activating for against specific things. And again, once that T cell sees, oh my gosh, in this lymph node or in the periphery or wherever the T cell is, oh my gosh, I see Deontay's eye and I'm activating. First things first, I'm the one cell that can see Deontay's eye. And so I have to multiply because I'm one cell. There are not hundreds and billions of cells that can see Deontay's eye. There's one cell only. I mean, literally, okay? So that one cell that can see Deontay's eye sees Deontay's eye, and it has to multiply first, so proliferate. Then those new anti-Deontay's eye cells, T cells, must differentiate. And then we'll talk about that in chapter 16. But yeah, so we can't do that without the monocytes helping out. All right, so here's a little question about it. Select all that apply, please. Which of the following cells are granulocytes? Oh, I have to click it. Here we go. Granulocytes. I think it's just one of these. Yeah. I had to read my choices.
Oh.
Yeah, that's me trying to be tricky. All right, so it's just one of these. It's just the fills. So monocytes, macrophages. Remember, macrophages are derived from monocytes. We basically have the monocytes and we have the lymphocytes. Those are the types of agranulocytes. We have subsets of them, but the fills and the mast cells. All right, so let's get moving on. The mononuclear phagocyte system. This is basically like extracellular network that cells can travel along. That's really all it is. It's like a highway outside of the cells to allow cells to migrate between tissues and within tissue, outside of the cells. Okay, so monocytes. They're monocytes because they have one big old fat nucleus. Okay, and they can turn into macrophages and dendritic cells. They're really important for phagocytosis. Many of our cells can also produce cytokines. So anytime that I recognize something foreign, whether it's a specific thing or a very generic thing, I see that this is a person. I saw a face. I don't know who it is yet. Our body doesn't know who it is yet. We saw a face. We know it's a person. We'll send out signals. The signals that we're sending out are called cytokines. This includes things like histamine. Histamine is a cytokine. So anything like that that can signal other cells to do other things, cytokine. Cyto being cell, kine is signal, okay? So there's 4 types of cytokines. Pro-inflammatory, they lead to inflammation. Anti-inflammatory, they tell the body, hey, we don't need to be inflamed anymore, we're good now. Vasodilators and vasoconstrictors, we increase or decrease blood flow. Again, you often want to increase blood flow to an area and decrease blood flow out of an area whenever we're first responding to a thing, but we need to reverse it in order to normalize things afterwards. And then growth factor cytokines. Growth factors are needed in order to trigger our lymphocytes to proliferate. So that's the idea with that stuff. So those are cytokines. And I'll talk about cytokines a lot, but luckily in my class, you won't need to know specific cytokines, but you may have heard of some of them. Things like interleukin or tumor necrosis factor or interferon. All of these are technically cytokines. Okay. So phagocytosis, we talk about it a lot. It's whenever a white blood cell eats up a bad guy, right? Phago being eating, cytosis is the cell doing the eating. We often do this in response to a PAMP. So again, we see endotoxin, we see peptidoglycan, we see double-stranded RNA, and there's other ones as well, that those basically monocytes or neutrophils or macrophages will see those things and say, this is a base, it's not self, so I'm gonna eat it now. So it's a way that we know it's not self, but we don't know specifically what it is yet. Okay, so our 3 major types, this is not an exhaustive list, but our 3 most important phagocytes: neutrophils, monocytes, and macrophages. I don't know what's going on with this. All right, so neutrophils are often easily the most important initially. Okay, so initial response is always the neutrophils. And you guys probably learned if you've taken anatomy or if you've taken physio, you definitely learned this. That like somewhere between 50 to 60% of your white blood cells should be neutrophils. They're the most numerous by far of your white blood cells in your blood, okay? And so they are very nondescript. They'll go out and eat anything that is foreign. Again, if it has a PAMP, we can see that it is a non-self, then we can eat it. And they'll eat it and they often die, or they will leave behind the waste after breaking something down. And so that's that process of phagocytosis. We'll talk about the steps of phagocytosis in a moment. Eosinophils can do this as well, but it's usually in response to different types of signal molecules, often related to parasites. And if you don't have parasites, then your body wasn't trained appropriately to trigger your eosinophils appropriately. So we can often trigger our eosinophils in response to allergies. If you, as we learn in chapter 17 in a little bit, you'll see third world countries don't have allergies because they had their immune system was properly trained to deal with parasites. First world countries where we no longer have parasitic infections, your immune system doesn't know what to do with eosinophils, with a lot of times basophils as well, or like your mast cells will hold on to antibodies to things that are not parasites. And so those systems were put in place to fight against parasites. And so eosinophils is part of that too. So we usually don't see eosinophils being elevated unless you have allergies. It's the number one reason in a first world country like the United States, or if you have parasitic infections. So if we don't think you have allergies and your eosinophil count is super high, you either have cancer or you have a parasitic infection.
Jesus.
Like that's your choice. Like those are, so not any of it good, okay? All right, so they can be phagocytic, but they're not our major phagocytes. Neutrophils, anytime you have any type of infection, you're gonna have high neutrophils pretty much. And so they're more general responders. So that's why we focus on them a little bit more. Monocytes are in your bloodstream. Once they get into the tissue, they turn into macrophages or dendritic cells. They are also going to be phagocytic. They also have lysozymes and things like that can help break things down and digest them. They are scavengers. So they're going to come in. Neutrophils are our first responders. They're going right into the fray and eating up the bacteria and things like that. And macrophages are kind of just like scavengers. They're picking up what the neutrophils left behind. They can eat active bacteria too, but they're usually there to pick up the parts and like collect all the parts and then go ask lymphocytes, do you recognize any of these parts? And that's really what macrophages do. Neutrophils can't ask the T cells anything. They just eat things non-discriminately. They're not even really like they're saying, hey, we have a fight going on down here, but they can't even call in the bombers or the tanks. Only the macrophages can call in the bombers and the tanks. And so they're coming and cleaning up and saying, do we need to call bombers or tanks now at this point? So the macrophages will be deciding that. You see how I choose bombers and tanks for B and T cells on purpose, okay? All right. When we are eating up a bad guy, again, you recognize a PAMP. First things first, how did you get called to that area? Because we don't just have like neutrophils just constantly camped out at places, especially like just sitting at normal healthy skin right here. If I were to take a cross section of my skin, do I have a lot of like neutrophils just sitting there? No. So how do they get to the area? They get signaled from the damaged cells in the area. So it's literally like damaged skin cells, damaged cells in this area, including mast cells, stuff like that does hang out in the skin areas like that in small numbers. But as soon as they pick up there's damage going on, they'll signal for the neutrophils to come in. And that's through those cytokines. So when we start sending out, hey, there's damage here, hey, there's damage, then the cells now nearby will be like, there's damage, there's damage, there's damage, all the way throughout your body. And the neutrophils in your bloodstream hear the call and they follow the signal to the area. That's chemotaxis. So I'm portraying it as me yelling, but they're chemicals. And they'll follow the chemical signals to the damaged area. That's chemotaxis. And they get there and they recognize a PAMP. This is a person that is non-self and I eat it now. So that's the ingestion phase. I come and I grab it and I envelop my whole cell around it and take it into a little vesicle. That vesicle is called a phagosome. We're tracking? Phagosome. I know you're looking at it and you're like, that's phagolysosome. Not yet.
Not yet.
Phagosome first. Then lysosomes that already existed in the cell, which is a little vesicle that contains digestive chemicals and things like that, merges with the phagosome, and then we break it down. Now, those chemicals are highly toxic. We're talking about reactive oxygen species, acid, and lysozyme that breaks down peptidoglycan, stuff like that. So lysosomes have lysozyme. It's true. Okay. So we're merging it with that, and it should break down pretty much anything. And so when that happens, that's called a phagolysosome. So it's forming that phagolysosome. That destroys the thing. Most of the time that takes 30 minutes. Within 30 minutes, we've destroyed a microbe or whatever you ate up in the area. And we excrete the waste. So this is what neutrophils do. They just eat it up, break it down, and excrete the waste, or they die after they eat enough things. They are not very long-lived once they start reacting to things. So that is phagocytosis. Again, a lot of that chemotaxis and ingestion is triggered by recognizing PAMPs. Cool. Here are PAMPs, again, peptidoglycan, LPS, also called endotoxin, found on the outer membrane of gram-negatives, and double-stranded RNA. The phagosome is the vesicle that's formed from the eating. The lysosome merges with it to make the phagolysosome. We kill the bacteria in 30 minutes. Here's a quiz about all that. What is a phagosome? Oh, right. Choose your choices. Be sure you read the choices carefully. All right, so it's the vesicle that is created. So the vesicle that is created from the eating, the vesicle that contains the chemicals for breaking things down was the lysosome. So the red choice, or sorry, the green choice on you guys' screen. The green choice is the lysosome. The phagosome is the red. When they merge together, phagolysosome. All right, when you have inflammation, these are the 4 major signs of inflammation. There's a 5th sign that's loss of function that doesn't have a Latin word associated with it. And we usually really only see that whenever you have a more injury going on with it. But the 4 signs of inflammation: rubor, calor, tumor, and dolor. Rubor is redness. I mean, they're related to the Latin word. So if you speak Spanish, this is easy for you, okay? But they're Latin words. Rubor is the redness. Calor is the warmth, which is where literally the word calorie comes from, right? If you didn't know that. However much energy is made from like how much energy you have to put into something to like steam like a gram of water or something, that's a calorie. And so that's 1 calorie. And what we typically call our food calories is actually kilocalories, thousands of calories. And so it has to do with energy that boils water, basically. But it's heat. That's a measure of that. Okay, so heat is the measure of that. So again, rubor is redness, calor is warmth, tumor is swelling, and dolor is pain. All right. It's all going to be made in response to injury. If I stub my toe, bam, the only reason it turns red and gets warm and swells and has pain is because of the signaling molecules that are being sent out from the damage associated with that. So if we're not sending the signals, you won't get any inflammation associated with that. That doesn't happen because of just rupturing your capillaries. It happens because we're sending in cells to repair damage. We send the cells in with fluid associated with them. All right, so chronic inflammation is associated with some diseases, including cardiovascular disease and autoimmune diseases. That can lead to prolonged chronic inflammation, can lead to damage in many of our systems, including your vascular systems, in your lungs, in your skin, and your joints as well. So we don't want chronic inflammation. If you're stressed a lot, you are signaling your body to have chronic inflammation as well. Believe me, don't ask me how to manage stress because I am also having problems with it. I wish I had the answer, but it's true that it can lead to inflammation in your body, increased inflammation. Why do we have inflammation? Number 1, it tells your body that you have damage. It's how you can know, hey, I must be hitting my shins a lot on things because I'm getting bruising and stuff like that. But it also, yes, question.
Those people that don't have that ability to tell when they're injured, does inflammation things still happen to them?
They'll have inflammation, but they just won't have that pain response. Yes. And so that can be an issue too, because if you're not getting pain for putting your hand on a burner, you don't know to move your hand away from the burner. So you'll just continue to cause damage. But they'll still have the inflammation and stuff like that. Yes. All right. So chief purpose of inflammation is to mobilize and attract immune components to the site so we can start healing. It's for the whole healing process. So we need to move signaling molecules in there to say, hey, we need to heal things. And then the cells will come in following chemotaxis to the area. We'll also have things like fibrinogen and other clotting factors in there to help clot any rupture red blood vessels and things like that. And repair the tissue damage as well as destroy microbes, which is the whole point of our immune system stuff. So here's another question. Select all that apply. Which of the following represent helpful functions of inflammation? We often don't think about it being helpful, but we just talk about some issues of them. What does inflammation do for us? Right, so this is all of these. Yes, so it begins cascades that destroy microbes, meaning that we're signaling in for things like neutrophils and macrophages to come to the area, and then they will recognize the PAMPs and start eating the things, but the inflammation triggers, it's part of it, the cytokines, the signaling calling for them, that's part of that inflammatory response. So again, you have injury and introduction of things into the area, and then cells in those areas will start releasing mediators saying there is injury, and we need to start reacting. Then the vascular reactions happen, vasodilation to get blood into the area and vasoconstriction to keep blood from moving out of the area. And then neutrophils move in by chemotaxis. And the fluids that come with the neutrophils, including all of the cytokines, as well as nutrients, things like that for the cells that are doing all this work. That fluid and your neutrophils, as well as any bacteria that might have gotten into the area, that is the primary component of pus. So pus is actually largely neutrophils, actually. Yes.
All that fluid is coming from our capillaries being penetrated, right?
Yes, so they're moving, it's moving out of the capillaries, whether they're damaged or not, it'll move out of the capillaries, the fluid will. Again, it's usually, unless your capillary itself is damaged, it's usually not actually blood. It's like the plasma that's also in there. So the white blood cells move between the cells and into the tissue. And that is a process called diapedesis, which is coming up here in a moment. But yeah, so the cells move in there, they start eating everything up. That's pus. And then finally, the macrophages come in and start cleaning things up and start signaling our T and B cells, and they can actually clean everything up. Pretty much the whole reason that after you have seen like an infection or something like that, the only reason that you can get better is because you finally activated and basically brought in your T and B cells, your tanks and your bombers. Okay. All right, vascular changes. We have increase of blood flow to areas and decreased blood flow from it while we are having that response going on. This is mediated by all those cytokines telling our capillaries and our arterioles and venules to basically, increase or decrease blood flow in the area. Exudate deals with the fluid that's come out of the bloodstream and into the tissues. That leads to swelling that we call edema. So edema is the swelling that we see in the tissue as a result of that fluid coming into the tissue. So exudate is the fluid. The edema is the swelling resulting from the fluid. All right, and that movement of the white blood cells through, again, you don't need to injure the capillary for them to be able to move into the tissue. They can squeeze in between the cells of the capillaries through diapedesis, okay? Diapedesis, and I think of pedesis of being like ped, like feet, like they're walking, they're moving. So that's the idea of how our white blood cells get out of there. And again, in the blood-brain barrier, these are tighter junctions and there's fewer of them. So we get fewer. There. All right, what is the term for the mechanism that enables white blood cells to migrate through the vessel walls and into the tissue spaces? I think we're almost done with this chapter. I think we've got complement left. We've got fever and complement, and then we can rock through chapter 16. Yes. All righty, so diapedesis is excellent. All right, so what's the benefit of edema and chemotaxis? Again, we're able to get our white blood cells and other signaling molecules and the nutrients that they need, as well as clotting factors, into the tissue. Again, this is going to be extracellular. This is outside of the cells. This is in that tissue spaces. It can also dilute any toxic substances that are there. We have antimicrobial peptides and things like that. We have clotting, and we have the neutrophils themselves to destroy the bacteria. In long-lived inflammatory reactions will eventually attract the monocytes, including out of the bloodstream, coming into the tissue. Now they are macrophages or dendritic cells. And what dictates whether it should be macrophage or a dendritic cell? The signaling from the tissue in that area, okay? And we'll get those guys in there with our lymphocytes to eventually kill things. Now, T cells, they'll have their function that we'll talk about in chapter 16 more, but the T cells are either captains that direct everybody, or they kill infected cells directly. The B cells produce antibody. That's all they do. The B cells themselves don't engage in the fight. So that's why I say the B cells are the bombers, because the bombs are the ones doing the work, not the bombers. The bombs are doing the work. And what are the bombs for our B cell? Antibodies. Okay, so we'll talk about that too. So that's coming. Fever. Fever is part of your inflammatory response. Once we start sending out all these cytokines, sometimes certain markers, certain PAMPs, or even certain immunogens specifically, can trigger fever. So that could be things that are outside of the body, but also if we get enough signaling going on between cells, cells can signal to make a fever depending on what's going on. So fever can be triggered either exogenously from things like peptidoglycan or endotoxin or other toxins and things like that, or endogenously from our own cells saying we should raise the temperature. What is a fever? Typically for an otherwise healthy adult, this is our range of fever. If you're, again, my husband comes to me sometimes with this 99, that my temperature is 99, I'm sick. BS all day long. You can go for a walk and get your temperature up to 99, like please. So a doctor will look at you and not roll his eyes visibly, but on the inside, his eyes are rolling if you come with anything less than 100. So it needs to be at least 100, okay? That's a fever. And are there times when a fever might appear when you are not sick? Yes. All right, but typically speaking, otherwise, if you weren't running out in a marathon or something like that, if you have a fever, it's the #1 predictor of there being some sort of immune response.
Would
Yes.
you ask this question from the test about like a geriatric patient's temperature?
I won't expect you of that. You will need to know that for nursing, right? So you'll need to know about geriatric patients and the difference in temperature whenever you go through the nursing program, but I won't ask about that on my exam. I do bring it up in class because you need to have perspective. So again, 98.6 is what we consider normal average temperature for the average adult. But many geriatric patients, you have somebody who's like 95 and is not really like mobile and is pretty much like a sedentary older person, the 98.6 might actually be considered a fever in that type of population. So we have to have perspective on those populations, but I won't ask test questions about that in my class. You will get asked questions like that in nursing school though. All right, so again, there is a reason for fever. Again, you can be signaled from outside sources like viruses and bacteria, from endotoxin, or from endogenous things like your own body decided like, hey, we've got a lot going on here, we should have a fever. All right, multi-select. Which of the following we would consider exogenous pyrogens? Exogenous. Hopefully I made this clear enough. Again, I'll help you. Exogenous is outside. Endogenous, it signals from inside the body. So, you know, your own cells signal for it. So endotoxin can signal for your body to make a fever. That came from not your body, that came from the organism. So endotoxin would be a pyrogen that would be exogenous. So then pyrogens are the molecules that trigger fever. Pyro, think of fire, think of heat. That's where the word comes from. What's the benefit of fever? It inhibits the microbes and stimulates our own body to respond. So fever impedes the microbes from growing from the high temperature. It slows down their enzymatic reactions, and it impedes their nutrition. It like basically decreases the availability of iron, which is a nutrient that they need. And it increases our metabolism. It literally speeds up our cells. It increases hematopoiesis, so the making of new white blood cells. Phagocytes act better. We have adaptive immune reactions as well as helping as signal our lymphocytes, our B and T cells to the area where they need to be. So that's all benefits of fever. Again, another question, I'm sure, benefits of fever. What are some benefits of fever? Select all that apply. All right, all of them. So yeah, this was an all of the above thing. All right, why treat fever? Should you treat fever? If it is a slight to moderate fever, generally speaking, it might be better to let your body run its course. If you're extremely uncomfortable or you have a high fever, then maybe treat it. And of course, if you have a kid, talk to the doctor always, like always talk about that. What's the safest drug to treat fever? Tylenol. Acetaminophen, yes, acetaminophen. So acetaminophen is our trade name for it. Yes, or our drug name for it. Tylenol, paracetamol are going to be the actual trade names for them. Okay, so treat it if you need to. Talk to the doctor if you have a kid. All right, interferon. Interferon is basically a signaling molecule from viral infections. If you're infected with a virus, you can signal your nearby cells that there is a viral infection going on inside of you. So it's basically like if I am living in my house and somebody breaks in and I don't have time to do anything other than hide, I can hide in my closet and call my neighbor and say, somebody's breaking in my house. You might want to grab the shotgun, Bill. So my guy next door, Bill, grabs his shotgun and then prepares for the guy to come to his house next. So that's kind of basically what we're going on, what's happening here. So interferon is the call to the neighbor. Okay, so it's a signal to the neighbors. Yes.
Yeah, I got confused because I kept thinking it was introns that we have turned off, right?
Oh, right. Introns. Yeah, it's a different thing. Interferons. So think of interfering. You're interfering with the viruses wanting to take over your cells by calling the cell next to you. Now, can it completely stop viral infections? Not really. It's not that effective, but can it slow the progress? Yes. And it's actually a pretty useful molecule for cancer as well. So we can actually use this as part of, in tandem with other medications, a way of telling your body to like, hey, be ready to kill some cells that are something's going wrong inside with them. So we can use that. All right, complement. The complement cascade. The complement system is a network of over 50 proteins that work together, and they're always in your bloodstream. You don't have to wait on your body to make them. They're always there. They're there right now. And they're usually the first molecules to act in response to things. So these are not cells. These are just proteins that are chilling in your blood. These are not cells doing anything, but they can signal cells to do things when they get activated, and they can come together and work as well. So it's a cascade, meaning that if one of them gets activated, it will break, and then those guys can like activate other guys, and then a bunch of them can activate, and so on and so forth. It's a big cascade reaction in response to things. They can get activated 3 major ways, and you do need to know the different ways. So the classical pathway is the pathway where antibody activates the complement cascade. So if antibody binds to something, it can activate the complement cascade. Antibody has a lot of different functions. That's one of them. Okay, so activating the complement cascade. Lectin. This is a molecule that binds to mannose. Mannose is a sugar found on bacteria. So if lectin binds to mannose, complement recognizes that, and the complement cascade begins. Alternative pathway. It's usually repeating molecules like PAMPs. But again, PAMPs are recognized by cells. So these are like PAMPs, but these molecules recognize them. Okay, so complement recognizes something like a PAMP. This would be LPS, peptidoglycan, same stuff, okay? Like PAMPs. They will recognize that, and then that can activate the complement cascade. So it just depends on what's activating it. Antibody, lectin binding to mannose, or PAMPs, basically, okay? All right, a little quiz about that. Which complement pathway is triggered by antibody? All righty, so that's the classical pathway. There's 4 stages to the complement cascade. Initiation, whenever you see lectin binding to mannose, whenever you see an antibody bound to something, that's initiation. Then we have the amplification and cascade. That's a result of the initiation. Polymerization is whenever those activated parts of complement start binding together to do their actions. There's several actions that they can have, including the membrane attack complex. Membrane attack complex, basically we have all these parts coming together, and they build and polymerize in a big old hole and make a hole in the surface of microbes. There's a video for this that I don't have time to show it, but it is a video that you guys are free to watch. It's a 7-minute video. We don't have time for it, but yes. Okay. So that is chapter 15. It is 10:30, so. So we're going to do this one. We're going to try it as quick as we can, okay? I want to just jump into this. What do you mean upgrade? Because
Maybe start again.
I'm just going to get out of it and just reopen it. Okay, we're going to rejoin. Here we go. Lecture. All right, different pin number 7592877. It's not gonna be super long in lab today, so I don't mind going over like maybe 10 minutes, but I don't want to keep people here forever, so.
We have lab right after.
Yeah, I know. It's just, I hate eating into lab time. This is probably our busiest lab that we're gonna have. Just saying, I don't want to have to make it rush either. But chapter 16, right? So we're gonna go ahead and get started. Again, the pin number will be at the bottom of the screen. So if at any point you didn't get joined into this one, there will be time. Okay, learning objectives. Once again. Sorry. I tried to give some warning. It really wasn't meant to be that. Okay, so this is the 3rd line of immunity, right? So now we're out of the 1st and 2nd in our innate. We're in the 3rd. So this is our B and our T lymphocytes. And this starts getting into immunocompetence, which has to deal with being able to identify foreign substances, specific Not saying this is non-self, but now being able to differentiate between our foreign substances. These all rely on specificity and memory. So they're highly specific to an antigen, which is a part of our bad guy. And we will lead to memory, which is basically the ability to react faster the next time that we see the bad guy. Antigens are also known as immunogens. These are basically foreign parts of a foreign invader that we can see as being a specific part. So they're basically molecules that can be identified by our immune system, but they are specific to the pathogen. So again, Deontay's eye, not just we now know it's a non-self. We can actually say, this is Deontay's eye. Okay, so that's the idea here. Very specific parts of things. They are protein or polysaccharide, but they can be any molecule that we could potentially react with. You must be able to perceive it as foreign and not as self. Your body spends quite a lot of time training not to react to self. So we have to be able to tell the difference between them. Most of our antigens, the larger they are, the better they will be, and the more like unique aspects there are about them. So things that are like one repeated thing over and over and over again, that's just like plain, not very good to react to. Yeah, so we want a lot of variability, and it could be any molecule that has that variability. Again, usually their chemical composition, proteins and polysaccharides are the best for the composition of them. And that basically can dictate what types of cytokines basically are being made. Like, let's say a neutrophil gets into an area and it's like, I saw, endotoxin, it'll make certain, cytokines in response to seeing something in a specific place, whatever. And those can actually help with our ability to identify our antigens as well. So the context, the chemical composition of the antigen, and their size. Generally speaking, larger antigens are better, generally speaking. The part of the antigen that you react to is called the epitope. So if you have a COVID spike protein, the part of the spike that we're actually binding on is the epitope. So the spike might be the antigen, but the part of the spike is the epitope. And so I often think I usually have fidget spinners, but I did not bring any today. But like, it's like basically like saying like I have this little keychain thing that I have with two little guys on here. The guys on my keychain might be the antigen on the whole keys, okay? So the guys might be the antigen, but the antibody only recognizes the feet. That's the epitope, okay? So the epitope is the part of the antigen that we're actually binding onto, okay? Haptens are things that are too small to elicit an immune response and need a carrier group in order to react to them, okay? This, and really good example of this is the tetanus toxoid. We cannot react to the tetanus toxoid by itself, but we link it to a carrier molecule or to other molecules. And now suddenly we can react to them. So tetanus toxoid, diphtheria toxoid, and pertussis toxoid, all too small, but we put them together and now we have parts to grab onto and interact with. So now when we see that part later, it's like during the training process of your body, we need the carrier. But then later on, once we've already activated the antibody to make to it, we don't need the carrier anymore. Make sense? Okay. Okay. So haptens are too small. This is an idea of a hapten, too small for the mouse to react to, no antibody, but we link it to something else. It's now big enough for our antibody to react to it. All right, here, pen and answer. What is the term for the part of the antigen that is recognized by lymphocytes? What is the term for the part? Gonna be real stinky in lab today too. So epitope, the part of the antigen that we react to is the epitope. Antigens that are too small will be haptens, but the part of the antigen that we react to is called the epitope. Okay, alloantigens. These are antigens that are associated with other people that help us see other people as foreign. And I mean this literally, not in a metaphorical sense, okay? So the reason that we need to have certain blood types, like I'm an A and I can't receive B blood, for example, because that's an alloantigen. I see B as foreign and my body will attack it. So we'll talk about that a lot more in chapter 17. Okay, so alloantigens are markers that we can see as foreign in other people. And this is the whole basis of immunocompatibility with transplants. Okay, and one of the biggest alloantigens that we can talk about is the MHC complex. There's super antigens as well. These are antigens that are very, very, very potent, and they actually cause overreaction of the immune response. So this can be things like endotoxin. Endotoxin is a very potent super antigen. Many bacterial toxins in general, not just endotoxin. Even gram positives make certain types of toxins that trigger a similar response. So those are our super antigens. Super potent response, overreacting of the immune system. All right, so now we're into the antigen presenting cells. There are only 3 types of antigen presenting cells that you need to know, okay? Macrophages, dendritic cells, and B cells. So remember, macrophages and dendritic cells were monocytes that went out and could interact with things, okay? So the monocytes and their derivatives, and then B cells. B cells, okay, they're on their own. All right, these are antigen presenting cells. I want to be very clear about this. Neutrophils are not antigen presenting cells. Are neutrophils phagocytes? Yes, they can eat things up and digest them, but they just eat them up, digest them, and move on. But these guys take eating up and digesting to the next level. We're going to eat up and digest and then also call out for help. Now, we learned that B cells, because we learned what our phagocytes were, right? Our eat up and digest cells. Those were the neutrophils, the macrophages, the dendritic cells, and monocytes. B cells are not phagocytes. So how are the B cells picking up antigen in order to present it? We'll talk about it, okay? Okay, you present antigen on either MHC1 or MHC2. Okay, we're going to talk about them, all right? Because people get so confused about these, and you're valid in being confused. All right, so we're going to do a question first, multi-select. All but apply. Which of the following choices can act as antigen presenting cells? Which of the following are antigen presenting cells? So one of these, I mentioned that they can act as a antigen presenting cell. They weren't on our list necessarily, but the derivatives of it were. All right, so this is monocytes and their derivatives. So remember, B cells and dendritic cells are monocytes. They're just in the periphery, okay? They're not in the bloodstream anymore. So monocytes still are going to be in the list, okay? But not neutrophils, so I'm glad we got that one. Good job. All right, major histocompatibility complex, MHC. There's 2 of them that you need to know. There are more than 2, but the 2 that I'm going to test you on, okay? MHC1 and MHC2. It stands for major histocompatibility complex. We also call it the human leukocyte antigen, or HLA. The lab that I worked at Baptist for the transplant lab, we called it the HLA lab. It's basically based on these markers. Now, these are really important in the immune response. They act as an arm to hold out specific antigen. It's really all they do. They don't care what the antigen is, but they say, I found antigen, here it is. It must be on the arm in order for our T cells to react to it. Okay, so that's the MHC. And then the T cell recognizes the specific antigen presented by MHC. However, my arm looks like my arm. Your arm looks like your arm. So my body will attack your cells because they have your arms on them. And my body sees that as like, why is there an arm holding weird stuff? And my body will attack it. So when we do cross matches for people getting transplants, we need to be sure their MHC molecules look the same as the person receiving the organ, because that's the thing that does the presenting of the antigen. Even though it might not be presenting antigen, it can just be chilling. My body sees your MHC as foreign, bad. We attack it and we kill your cells that have it. All of the cells in your entire body, if they have a nucleus, they can have MHC1. So MHC1 is the molecule that we use to say I'm infected inside with something. So obviously all of your cells need to be able to say I'm infected inside with something. So again, to me, MHC1, the 1 looks like the letter I, like inside, okay? MHC2 molecules present things from outside of the cell. So in order to get something from outside of the cell, we often have to eat it. And so what cells can eat it? Phagocytes. But not all phagocytes have MHC2. Only our antigen-presenting cells do. So again, dendritic cells, macrophages, and monocytes are phagocytes that can eat things up and present on MHC2. B cells. B cells interact with antigen using the B cell receptor. It looks like an antibody, but it's on the surface. Okay? That antibody recognizes the thing on the surface of the cell, picks it up, and says, oh, I picked up the thing. I see Deontay's eye, because it sees a very specific thing. And so I see Deontay's eye, and it takes it inside because of that, and then puts it on MHC2. Okay? So if we picked it up from outside, either through phagocytosis or through the B cell receptor, we take it in and then put it on MHC2. That's our way of saying we found this outside. Otherwise, if you're infected inside with a virus, and that virus is growing inside of you, and you're like, oh my gosh, there's something inside of me. What's going on? Help, I found this inside of me. MHC1 does that. Are we tracking? Okay. Please, because it doesn't get easier from here. All right, so that's the idea of MHC. MHC1 is on all nucleated cells. Cytotoxic T cells recognize antigen that's presented by MHC1. Cytotoxic T cells. So if I'm sitting there saying there's something, I had a face hugger on my face. Now I have something growing inside my chest. Help me, help me, help me. This is what I found growing inside of me. The cytotoxic T cell, the angel that it is, comes and says, yes, I see that, and kills the cell. And that's what cytotoxic T cells do, okay? But that's dangerous for your body to be able to have T cells, again, cytotoxic T cells, go around killing self cells. That has to be very regulated. Otherwise, your cytotoxic T cells could be killing your own cells constantly. I want to be very clear about this. You guys have heard of diabetes. Yes, type 1 versus type 2. Okay, type 1 diabetes is caused by cytotoxic T cells killing insulin-producing beta cells in your pancreas. So that's an autoimmune reaction. So you can see how out of hand that can get just from one type of cell being targeted. Now, in a transplant, my body sees your kidney cells, MHC1, and my body will kill those cells so fast, bam, 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 that you can be transplanting somebody's organ into them. And if they've seen that before and had a T cell response before, you can actually see the organ die while they're doing the surgery. It can be that fast. So this is really important that the T cell response be regulated. So there's two types of T cells, cytotoxic T cells and helper T cells. Okay, so we're going to get to that too. So MHC1 is recognized by cytotoxic T cells. But cytotoxic T cells, it's not like the cytotoxic T cell comes up and says, here's MHC1 holding up the antigen. Cytotoxic T cell says, yes, I see this antigen, this Deontay's eye. Okay, we see this and we know this and we've seen this. It can't just kill it. It has to have the go-ahead from the captain. That's the helper T cell. So the helper T cell needs to have seen this too. But the helper T cell doesn't see it in MHC2 or MHC1, excuse me. Helper T cell doesn't see it in MHC1. Helper T cells only see things presented by MHC2. And remember, MHC2 is what we present outside antigen on. So you can't have an inside response without having seen the outside first. Are we tracking? Okay. Here's a quiz about MHC1. What does MHC1 do? Two of these I think that are correct. Oh, it only lets you choose one. Okay, never mind. It's one of them that's correct. I didn't read the choices. That's my fault. Yeah, it's only one of them. You're right. It's just one. So again, presents inside antigen to cytotoxic cells. So cytotoxic cells are known as CD8. And this is how I remember this too, is because I remember the number 8 is a very round number. And the number 4 looks like a letter H to me. I mean, which one looks more like an H to you, an 8 or a 4? Me, a 4 does, yes, I don't know, if that confuses you, this is how I remember it though.
Color it. I just said, let's not do this.
Yeah, you don't have to if you don't want to, like I said, but this is it, you do have to know which one has 8 and which one has 4, and they are different, yes.
4281. 4281.
Yes, 4 goes with 2 and 8 goes with 1. However you want to remember it, again, you, okay? So, okay, so class 2, so we're gonna do a little bit of drawing on this because now we have that class 2 and class 1. Where's my drawer at? There it is. All right. All right, MHC. Type 1 binds with CD8. CD8 is found on cytotoxic T cells. MHC2 binds to CD4. CD4, and again, CD means cell differentiation marker. It's not anything fancy, but whatever, it is what it is, okay? I didn't choose this for you, okay? But CD4 is found on helper T cells. Again, both of these are T cells. Okay, we haven't even gotten to the B cells yet, right? Now, this is MHC and how MHC works. All right, what happens is you have a virus that is outside that gets eaten up by, I don't know, a macrophage or something. Okay, macrophage finds that outside, brings it in, displays it on MHC2. I'm going to put the letter, or no, let's not actually, let's put the number 2 because it's
Would
just
those
easier to see.
be the epitopes?
So we haven't gotten to the epitope yet. Right. So what I'll say is, this is let's say this is the virus spike protein. Okay. Here's the virus spike protein. Then we're going to have the T cell receptor recognize part of the virus spike protein. That would be the epitope, the part that we're reacting with. This is the T cell receptor, okay, that is found obviously on the surface of a T cell. This is a T. If we're interacting with MHC2, which T cell interacts with MHC2? Helper. Okay, so this is a T helper. All right. And literally what's happening here is this is CD4. It's a molecule on the surface of it, and it literally interacts with MHC2. So it's not like you have CD4 and that cell that has CD4 recognizes MHC2. The CD4 itself recognizes the MHC2, okay? CD8 on the cytotoxic cells recognizes MHC1. So if this was a cell that was infected inside, it's the exact same process, but instead of CD4 on a T helper, it's CD8 on a cytotoxic T cell. So I am activating my T helper first. This interaction here allows activation. Okay, so now we've activated our T helper cell. This T helper cell then goes through proliferation. It would help if I started with a P, proliferation. So proliferate into a bunch of T helpers. And then each of these will differentiate into a T helper that is a regulator, a T helper that is a memory. I should make 2 of these. And a T helper that is TH1 and a T helper that is TH2. These guys down here are our effector cells. These are the guys that are actually doing the jobs. Okay, so these are our actual captains. Now that they've been activated, now when a cytotoxic T cell interacts with antigen on MHC1, they'll get the signal from one of these T helpers to kill. Okay, so TH1, this is important that we know, TH1 sends signals to CD8. Response, Th2 sends signals. Come on, dude. I'm clicking it. The B cell response. Okay, this is just the T helper response. You must activate the captains though. So we cannot run our CD8 response to kill inside infections without the Th1 captain. Okay, and we cannot signal antibody production. We cannot signal antibody production without Th2 captains. Okay, so all of this relies on T helpers. T helpers are signaled from MHC2 that is only found on antigen presenting cells that pick up outside antigens. You can't have an inside response without an outside response. Now I will remind you again, having shown you that HIV infects CD4 cells. So we eliminate all those captains, all of them. No Th1 and no Th2 if you have HIV. That's a huge problem, right? Okay, so we tracking this. We have the helper cells down. Okay, MHC, again, I'm a macrophage. Somebody stubbed their toe. Mast cells and injured cells in the area call out for help. Macrophages make their way to the area and start cleaning up what the neutrophils ate up. I'm just skipping a step, but still, okay, like neutrophils got called there first. They're crapping out whatever they break down. Macrophages come in and start cleaning up, and they start finding stuff all around in the area and eating it up and being like, anybody recognize any of these on each of their arms? They picked it up from outside, so the arms holding it are MHC2. All right, T cell, T helper, CD4 positive T helper cell comes on, and it says that's Deontay's eye, alert and activates, proliferates, and then differentiates into all those different versions, including Th1 and Th2. So now we have Th1 cells and we have Th2 cells. Do you have more of one than another? Potentially, depends on the signals we're getting in the area, okay? So in order to control wherever that stuff is that got in the body, where I stubbed my toe, to control the outside response, we will trigger the antibody stuff with Th2. If we need to trigger inside response, that'll be dictated by Th1 helpers. Yes.
So is HIV picked up from the outside, put inside, and then it affects the T helper, or how's that?
So it doesn't have to go that way. It can quite literally, like any just T helper just chilling out, minding its own business. It has CD4 on its surface. It uses it as a receptor.
Okay.
Yes, so it's a completely separate interaction. Yeah. All right. We're leaving it up and moving on, okay? Here's a question. Which of the following choices applies to MHC2 molecules? Sure you're reading your choices here. I have it written up here in 2 different places, but. All right, so MHC2, again, to me, MHC1, the one looks like an eye, that's our inside response. MHC2 is our outside response. So we pick up stuff from outside. We present outside antigens, all right? Then it's up to you to remember CD4 and helper, CD8 is cytotoxic, okay? So this is our helper T cells. So MHC2 is recognized by the CD4 molecule on our helper T cells, which is happening right over here. Okay. You get 1 chapter for this. All right, and when I tell you guys that immunology is one of the most undertaught things, you can't tell me it's not now. And when you get into this class and we talk about this, and it's just like all Greek. All right, lymphocyte markers, we call them receptors. And so we already saw the T cell receptor over here. Again, the T cell receptor recognizes an epitope of the antigen. And that's how that T cell was only designed to see Deontay's eyeball. There's another T cell that might see his hair, but there's another T cell that sees Maddie's hair or Maddie's eyeball or Professor Ghost's hand or whatever. So you have T cells for all different types of antigens and epitopes, okay? So those things that interact with the antigens and epitopes, those are the T cell or B cell receptors. We've seen images of a B cell receptor before because we see them as antibodies. Antibodies are the same thing as the B cell receptor. It's just if it's membrane bound on the surface as a receptor, then that is the B cell receptor. If it's made and secreted, then it's the antibody. Okay. So this is where we first see our antigen as a B cell. B cells usually hang out in the lymph nodes. And as we're filtering, and you stub your toe, we're filtering out that lymph. They're waiting in there, checking all the passports of all the molecules that come through. They see a molecule come through and they say, I was a B cell checking everybody's passport, just looking for, again, Deontay's eye. And I'm a B cell that sees Deontay's eye. And my antibody grabs onto the eye and takes it inside and puts it on MHC2 because I found it outside. Okay, so that's how it will interact with that stuff first. This is as the B cell receptor. We know that, again, it finds that, puts it on MHC2, Deontay's eyeball hanging out here on MHC2, the T cell comes and interacts with it, and they can activate one another. Once the B cell is activated, it proliferates many B cells. Some of them become regulators, some of them become memory cells, and some of them become effector cells. Effectors are the ones that do the job. B cells that are doing their job make antibody. B cells that make antibody, we call them plasma cells. And so all they do is crank out soluble versions of that antibody. It's the B cell receptor, but now it's a soluble version of it, okay? T cell receptors, they're similar in how they interact, but they don't ever get secreted. They're only ever on the surface of the T cell. Are we tracking? Okay, it's a lot. All right, antibodies, also known as immunoglobulins, okay? And you've heard of immunoglobulins because people have to get immunoglobulin infusions because they can't make their own antibodies. So this is an immunoglobulin. The top part at the top of the Y, that's the variable region, and that's the part that interacts with the antigen, okay? And so it's variable, that's how we get all those different variations. And that's where we also have the gene reassortment going on. There's a video that goes along with this that I'm not gonna play, but you're welcome to watch it on your own time. The bottom part, the base of the Y, what's basically attached to the surface of the B cell receptor, or it's just the bottom region of the antibody, that's the constant region. And it dictates the function of the antibody. So it dictates whether it's gonna be membrane bound receptor or whether it's gonna be secreted and does these various jobs, okay? So that is the constant region. Variable and constant regions. T cell receptors are a similar type of shape and have similar type of variability and are actually like a gene reassortment the exact same way, but they are just always going to be bound to the surface and never secreted. And they're a little bit smaller. All right, so here's that idea. We have all these different choices of genes you can put together and then reassort things in the middle, add and delete nucleotides, and then get these different versions of our immunoglobulin. Again, here's the video.
Of antibodies.
It's really
A
cool.
lot of antibodies.
So I do recommend you watch it. It's made by Nature. So it's a pretty cool little video there. Okay. So you can make 8.5 billion different cells that can react to 8.5 billion different things, but some of those are anti-self. So we have to train your body to basically like, hey, you made a B cell against your own body. We need to kill it. We can't keep those guys around. That training happens very early on in life. And so this is the idea of clonal selection. We delete clonal deletion of anti-self clones. And then whenever I see Deontay's eyeball, I only proliferate the B cell that sees Deontay eyeball. I don't proliferate B cells that see Lauren eyeball or Taylor eyeball or anything like that. Only the one. So whenever you activate a T cell, you're only activating that T cell to that antigen, not all of your T cells. So that is clonal selection. Okay. There's a little infographic for it. We have to tolerate self and react to non-self. B cells often will basically home to sites in lymph nodes and hang out there. They have immunoglobulins as surface receptors, whereas T cells cycle constantly throughout your circulation to try to find antigens all the time. It's way more important that we activate our T helpers. And then our cytotoxic T cells have to go out into the field and kill infected cells. So they circulate a lot more, whereas B cells usually hang out in the lymph nodes. Again, T cells, they do work. They're not making antibody that does work. The T cells themselves are doing work. And so our T helpers are basically creating cytokines that direct the immune response and help activate other cells, whereas cytotoxic T cells are actually actively killing infected cells. So those are cell-mediated immunity. Cell-mediated immunity has to deal with your T cells. We call B cell humoral. They're humors, the liquid in your body, but it's basically humoral immunity because the antibody is what is doing the work. Okay, so T cells have helper, cytotoxic, and regulatory, and also memory, right? But the memory is not doing anything except waiting, and then it becomes these when you reactivate it. So that's the idea here. So helper T cells activate all of our other immune cells, including telling macrophages to go back out and eat more stuff. So that's another thing they can do. But they basically direct everything. Cytotoxic T cells kill infected cells. The CD4 is found on the helpers. The CD8 is found on the cytotoxic. The CD4 on the helpers recognizes MHC2. The CD8 on the cytotoxic recognizes MHC1. If you're going to do anything for this chapter, know those, okay? Know that right there, okay? CD4 is on helpers and it interacts with MHC2. CD8 is on cytotoxic, it interacts with MHC1, okay? Definitely like start there. All right, true or false? CD4 interacts with MHC2. Yeah, that's why I don't base it on grade, man. I don't know.
I can't even click an answer. It's just
All right, great job. Beautiful. All right, so those T helper cells, again, they are going to be our captains. Our major ones that I ask about by far the most are Th1 and Th2. So as a T helper, you have your little CD4 recognize MHC2 and your T cell receptor recognizes the antigen. You got both of those happening when we interact with like an antigen presenting cell. Then you get activated and you turn into Th1 and Th2 and memory and regulator. Okay? The Th1 and Th2, the Th1 goes out and sends signals to activate our CD8 response and macrophages too. Our Th2 response activates B cells and making antibody. Yes, I think it is a like regulator in memory, but there's another video.
CD4 cells
We'll watch this one.
with foreign antigens attached to MHC2 molecules. They become activated. This activation results in the production and release of interleukin-2
The cytokine.
The released interleukin-2 cytokines come back in autocrine fashion to activate their releasing CD4 cells. This activation induces a cascade of intracellular reactions, ending with the replication of T helper cells to two new T cell lines, T helper 2 CD4 cells and T helper 1 CD4 cells. T helper 2 cells are the main line for antibody immunity, which is called humoral immunity, whereas T helper 1 cells are actively involved in cell-mediated immunity. Remember, interleukin-2 production, receptors, and intracellular effects are important targets for immunosuppressant drugs. Now, let's focus on antibody-mediated immunity. And later on, we will come back to T helper 1.
Like obviously there's a series of videos there, but anyways, so yes, so we have our antigen presenting cell. All right, so this is now moving on to the cytotoxic ones. So again, you have your antigen presenting cell. We have found something inside. So if we find something inside, you're an infected lung cell, you're an antigen presenting cell. It doesn't matter. Now we have an inside response. Inside is presented on MHC1, inside response. So now our cytotoxic T cell can see that, but we can't act until Th1 from our outside response comes in and makes interleukin-2 to activate that. Again, this is a major signaling molecule in a lot of our cascades. I won't ask about interleukin-2, but you should at least know that a cytokine and that there are a lot of different ones that basically help with all this signaling that's going on here. So again, a lot of our treatments like Humira and stuff like that, they target, like they block these signaling molecules. And so if you block it, it helps with your inflammation, but it also blocks your body's ability to fight infections, which makes you more susceptible to infections overall. So anyways, yes. So once we activate that cytotoxic T cell, they make things called perforins and granzymes, which perforins do what they sound like. They perforate, they poke holes in things, and they poke holes in the infected host cell. So if I'm sitting here being like, hey, I have a chest burster in me, help me. The CD8 cell comes and sees the antigen with its T cell receptor. The CD8 molecule sees the MHC1, and then the TH1 cell sends the signal to activate it. And now it says perforins and granzymes, pokes holes in me, and the granzymes digest the things inside of me. And that causes the cell to die. That's how it targets cells. So that's how the cytotoxic T cells work. A little bit more explanation on that if you need it. So here's our quiz. So you're fine. Which of the following are required in order to activate CD8? What do you need in order to activate your CD8? Hopefully you can, I mean, this is a picture up there, but I know it's a lot. I should have given more time, but you get 30 seconds. This one is going to be blue and green. We did pretty good. We didn't do terrible on this considering, okay? So it's blue and green. So you need activation signals from an activated CD4TH1 cell. Again, that was activated from an outside response, right? That captain. So this is the outcome of that captain, the TH1. The TH1 basically gives the signal so that the CD8 interacting with the MHC1, its T cell receptor sees the antigen, the CD8 sees the MHC1 molecule, and now we can make our perforins and our granzymes. But you need both of those. Interaction with the T cell receptor to the antigen being presented on MHC1. CD8 has to recognize MHC1, and we have to get the signal from our TH1 cell to give the go ahead. Both of those things have to happen. All right, perforins poke holes, granzymes digest things in the target cell. That often leads to apoptosis. The cell knows it's dying and will signal to just go ahead and die. This often targets virally infected cells, cancer cells that your body can recognize, and cells from other humans and animals. B cells now. Again, B cells are going to be able to release antibodies, and they're also going to need a T helper cell to signal the go ahead with that's this process down here. The TH2s are going to give them the signals to go ahead. Again, they can pick stuff up with their B cell receptor, but if they picked it up with the B cell receptor from outside, it comes in, then we present it on MHC2, that interacts with our T helper cell, and then we can give the signals to activate the B cell. The B cell will then go ahead and proliferate and then differentiate into regulatory, memory, or plasma cells. The plasma cells are the ones that make the antibodies. That's what these like snowflake things are here. All right, here's a quiz question. Select all that apply. What is required to activate a naive B cell? What do we need to activate that naive cell? All right, so this one's getting a little bit blurry now. So I want to be very clear. The B cell does not interact with MHC. Only T cells interact with MHC. Only T cells interact with MHC. MHC1 interacts with CD8. MHC2 interacts with T helper. That's it. B cells do not interact with MHC. T helper 2 cells made from MHC2 activating the T helper turned into TH1 and TH2. The TH2 gives the signal to the B cell that it can proliferate and differentiate and make antibodies. So we need that. That is the red, okay? The signal from the TH2 cell. We also need the surface receptor of the B cell, which is the antibody on the surface, to interact with the antigen. Okay. We could see that in the previous slide, which was here. Antigen is going to be interacting with the immunoglobulin on that microbe, presents it on MHC2 here, and now we can make antibody to interact with.
It's the antibodies in complement.
This blue one?
Yeah.
Okay, so in order to activate the B cell, so that's something that antibody can do after we make the antibody, but we have to activate the B cell
Okay,
first.
okay.
And then that is a result of making the antibody. Does that make sense?
Yeah.
That's a role of antibody. Okay, good question. All right, again, we're reintroducing that antibody. We have the variable region and the constant region. The variable region can also be known as the FAB fragment. I didn't make these rules, okay? This is the antigen binding fragment, antigen binding AB. C, thankfully, constant region. It's also known as the crystallizable fragment. I will never ask about that, but I think of C as being the constant region. FC is the constant region. FAB is what makes the antibody. It's the antigen binding part. Okay, so that's the variable region. We kind of already covered that. All right, so functions of the FC fragment. So we know that the FAB interacts with the antigen. The FC determines the function of the antibody, okay? So the constant region. So opsonization basically serves as a molecular handle for us to grab onto. So it's like all that sticky coating with the honey on the capsule. These guys can stick to that, and then we can grab onto the antibody now. It makes it easier to grab onto things. That's opsonization, all right? Some of them block, basically block the ability of viruses or bacteria to bind onto surfaces in the host. We can block the toxins that they produce. And we can signal for them to like cause certain cytokines to be produced and all sorts of stuff. So here are the functions. Oh, not quite yet. All right, what is the best choice for opsonization? What I call the molecular handles. All right, so they serve as molecular handles for our phagocytes to grab onto. And antibodies can do this, but so can complement. Complement can be opsonizing as well. So antibodies or antigens basically make it easier for phagocytes to eat things. All right, so here's the functions of antibody. This is opsonization here in the middle right here. Okay, serve as molecular handles so phagocytes can eat us easier. We can also just coat the surfaces of bacteria so that they can't reproduce or interact with each other. So that's the idea of that coating process. This is neutralization. We're blocking specific, like the spikes from docking with the ducts on the host cells. So specific blocking of the docking mechanisms. Agglutination, causing the cells, the bad guys, to stick together and clump together. And then we can eat up those big clumps. Activating complement, we learned about the classical pathway of the complement pathway. And then antitoxin, antibodies that bind to the toxins, like what we see with tetanus and stuff. All right, there are 5 classes of antibody. The immunoglobulin that was on the surface, the receptor one, that's IgD. Okay, that's the surface receptor one, IgD. IgM is the one that's always made first. It doesn't work as well, but it's what we can crank out immediately. And then as we're going through our differentiation and stuff like that, we will hone that in to these other types. How do we know to make IgG, IgA, or IgE, the other 3 types? Because we're getting signals and cytokines in our environment telling us which ones we want to favor. But generally speaking, we're always going to make the most of IgG, typically. IgG is our workhorse antibody, and it can do all of the antibody things. The weird snowflake IgM is this pentamer here. It can't do opsonization very well because we can't grab on to the FC region very well. So it doesn't do some stuff as well. The IgG, it can do all of the things, and we're usually going to make about, like, once you have an antibody response to something, 80% of it is IgG. So we have just a lot of IgG eventually once we start making it. But again, the first one is IgM. Then we will switch, class switching of the antibody. Once we start getting signals and we get things underway, we would make first IgM, then we'll switch to either IgG, IgA, or IgE. IgG is our workhorse. It's found in the bloodstream. IgA is found in the mucosa. So if you have an infection in one of the mucosa areas, IgA will be your buddy. IgE is made in response to parasites or allergies. Again, depends. Okay. We'll talk about how it works in allergies and stuff like that in chapter 17 next. All right, which of the following choices best represents the function of IgA? Wish that the letters went along with their functions better, but it's just the way that it is. And like I said, the review session is going to be me walking through all of this again, very stepwise. And so you'll have better reference and hopefully it will make more sense by then. But for now, definitely work on focusing that MHC1, CD8 cytotoxic, and MHC2, CD4 helper. Those are T cells only, and then focus on the B cell stuff. And then coming back to this after that, it will be a lot easier if you can get that stuff sectioned out. All right, we can monitor antibody production over time through titer. Titer is basically looking at the concentration of antibody in a patient's sample, whether that's in blood or something like that. It's usually going to be serum. You usually have the most antibody the closer you are to the exposure to the thing. Now, we all know that if you get sick with the flu, you're sick for a few days and then you recover, right? That whole time you're sick with the flu for like a week that you're sick, we're waiting on your B cells to make antibody. That's what we're doing. Or your cytotoxic T cells to come and kill infected cells. We're waiting on that. Once the cytotoxic T cells and the B cells are ready to go, we have our tanks and we have our bombers. Only once they're ready to go can your body actually fight off the infection. All that inflammation, that fever, that pain that you were in was your innate response. Once these guys kick in, it's like, you're done and you feel 100%.
That's our lag time right there.
That's the lag time. That's the lag thing. So it's the time in between. This is what we call the latent period for your primary response to something. Your secondary time that we see something, the second time, like with chickenpox, for example. So I had to go through having the chickenpox and wait on my cytotoxic T cells and antibodies to form. Now, when I see the chickenpox, I didn't even know my body ever saw it. I'm that fast. That's your secondary antibody response. And so that's your memory response coming back. Antibody is fast. We don't have to wait on proliferation and differentiation anymore. We just activate the memory cells and then just immediately do the thing. Okay, so that's the secondary. So again, here, you have your exposure to the antigen. We are waiting on your B and T cells to get activated. First, we make IgM, then we'll switch to IgG, and then we get rid of the infection. The next time that you see the thing, immediately can make IgG. You don't even have to wait. And you clear the infection so fast, it's like you never even knew it was there. Ideally, and most of the time, that's going to happen. Okay, there's a whole video on it. Again, we don't have time for it, but the Amoeba Sisters makes a great video explaining this. Okay, all right. Active and passive immunity. Active immunity is when you make antibodies to something. Passive immunity is when I give you antibodies to something. Okay, you didn't make them, I gave them to you. We have natural and artificial on both of these. Natural is this happens in nature. Artificial is medical community introduced it to you. So active immunity, you make your own antibodies. When I had chickenpox and now I have antibodies against it, that's natural active. If you got vaccinated for chickenpox and you now have antibodies against it, that is going to be active artificial. Make sense? Yeah. All right, if you are breastfeeding your baby and they get antibodies from you, that's passive. You're giving antibodies to the baby, but it's natural. But if you get immunoglobulin infusions, then that is artificial. Make sense? RhoGAM is an example of this as well. RhoGAM is going to be an antibody against antibodies, anti-baby antibodies. And we're going to talk about that in chapter 17 later on. The idea is that with vaccines, we can introduce something to your body without having to have the disease. And so we can tell your body, hey, make antibodies against this thing, and now you'll be protected so that you get that immediate response instead of having to have the disease and then get the response. So that's the whole goal with vaccines. There's different kinds of vaccines. We can give you the killed or inactivated whole virus. We can give you a live attenuated version of the virus or bacteria. This is something that doesn't cause disease anymore, but it might elicit a better immune response. We can also use parts of the organisms as well. So if we use parts, we could use the surface antigen. We can use mRNA that then makes the antigen, or we can complex the antigen with other things to make it better elicit a response. So those are the different types of vaccines. Booster shots are doses that just help keep your memory alive. Your memory does not last forever. Some immunogens or antigens trigger very long-lasting memory. Some of them it's shorter lived. It just depends on you and your body and your genetics and the cell and the antigen and all the signals that are coming with it. So the idea is that booster shots keep that memory alive. We can administer vaccines subcutaneously, which is just under that skin, intramuscularly, which is into the muscle tissue, or intradermally, which is different than subcutaneous. Intradermally is like where we will make the scratches or excoriation. on the surface. We see that mostly with the smallpox vaccine. We don't really see that much elsewhere, which we don't really give smallpox vaccines unless you're in the military. Oral or nasal vaccines, again, we don't see a ton of these unless you are allergic to eggs. You might see the oral or nasal flu vaccine this way because the injected flu vaccine uses eggs. But however, in other countries, these are usually more stable than the injectables, and you don't rely on having to have needles. So often in other countries, when we get vaccine efforts, we'll use oral or nasal vaccines because we don't rely on having to use needles in those cases. Polio vaccine, for example, is an oral vaccine. We see that a lot with dogs too. So if you're going to the veterinary field, we get, I think the kennel cough, which is a Bordetella type, that's oral. They'll just shoot it in your dog's nose. Anyways, adjuvants are things that enhance the immune response. So we add it in, like we can give you COVID spike protein, but we can give you some stuff that helps trigger the proper immune response to it as well, so that you get better memory and better response. For example, having measles can actually infect your memory cells for all types, different types of memory to all different types of diseases. But if I give you the vaccine against measles, I can make sure you're having an actual immune response to measles and you're not killing off other memory cells. So it's safer and more controlled that way. Is it perfect? Is there any side effects associated with vaccines? Of course there are. Okay, no doctor or scientist is ever lying to you about that. Of course there are issues associated with this. Common side effects will be local reactions just to the vaccine itself, as well as short-term flu-like symptoms to vaccines. This is your immune response reacting to the thing. If you see an antigen and your body triggers inflammation to an antigen, that's what we're trying to do with the vaccine. It's normal to have fever, soreness, malaise, things like that after vaccine. Do you always have that? No, it depends on your body. Depends on how it was delivered, when it was delivered, what other things were going on in your life, how much cortisol you had in your body, and all sorts of stuff like that that can predict that stuff. Are there other side effects associated with it? Yes. For example, anaphylaxis, which is also an immune response. Super rare, much, much rarer to have side effects that are serious than side effects associated with the diseases themselves in almost every single case of any vaccine that's ever existed. By far, like much, much more likely to have severe issues with having the disease than you would with any vaccine that we develop. So FYI, misinformation, we've mostly talked about a lot of this stuff, but again, can you get the flu from the flu vaccine? You can't. It's actually impossible. You don't get the flu from the flu vaccine. If you thought you did, yeah, I did. I got this, the fever and stuff. No, that was your immune response. You didn't actually have the flu, and you weren't contagious either, but your body had the immune response. 90% of any disease that you have where you start feeling sick and a fever and inflammation, that's your own body's immune system making those signs and symptoms, not the actual infection. So yes, so that's the inflammatory response. The other thing, mercury in the measles vaccine can cause autism. There's less mercury in a vaccine, and all the vaccines that you'll ever get in all of your life forever and always add up all that mercury, and there's probably more mercury in a can of soda. You want to talk about that? You want to talk about that? FYI, okay? If you want to talk about that, we can talk about that, okay? All right, so that is it. One last question, if you're still on. Vaccination activates a completely different type of immune pathway than natural infection. It's a different type of immune pathway. Does it still activate the B cells and the T cells and stuff like that? I would hope that it does, so this one should be false. It's not different. It's the same exact immune pathway that having the illness would activate. This one should be false. All right. Good job, guys. I'll see you up in the lab. Great job sitting through this. I hope this was helpful for you guys. And then at least now, if you watch the video and you aren't paying 100% to the video, it'll make more sense. All right. Any questions before we head up to the lab? It definitely is going to focus more on 15 and 16 and 17 a lot, but 17 needs 16. Yeah. So yeah.